Good afternoon, everyone, and thank you for joining us for today's webinar. We are going to get started here in just about 5 minutes, but before we do, I want to go over a few housekeeping items with everyone. Today we are utilizing a brand new platform. I know that some people have had some technology issues getting into the room. We do appreciate everyone's patience working through those. But the first thing I would like to make mention of is our chat pod, which is still located on the left-hand side of your screen. You should be able to chat in that the chat roll chat pod there. Should you have any questions regarding today's material, all questions pertaining to the material will be retained until the Q&A session that will be held at the very end of today's webinar. Or workshop, I apologize. So please feel free at any point of today's talk to go ahead and type in your questions there, and those will be housed, and then we will get those answered at the very end of today's workshop. This chat pod is also the best way to let us know should you experience any technical issues. If you do have any technical issues, please make sure to let us know there in the chat pod, and one of our many customer service representatives that we have on today's event will make sure to get you taken care of. All of your audio for today's workshop is going to be streaming through your computer speakers. So again, if you are not able to hear audio through your computer speakers, let us know there in the chat pod, and we again will be sure to get you taken care of. We've got about 4 minutes before we do get started. So we have quite a large group in today's event. We always like to find out a little bit more about our audience. So if you could take a few moments just to in the chat pod to let us know where you are watching today's event from. That'll give us some great information about our audience, and also will give you the indication that everything is working properly for you with the chat, and that you are able to type in any questions should you want to. I'm going to mute myself back just for a few moments, and I'll come on with housekeeping once more before we do get started, and then we will start here at the top of the hour. We've got about a 4-hour workshop, and we want to make sure that all of our presenters have ample time to go through their presentations. Again, thank you so much for joining us today. We appreciate everyone's patience. We hope that everyone has a great experience with our new platform, and we're going to continue to make adjustments as needed. I'll speak with you in just one moment. Thank you for joining us today. Good afternoon, everyone, and thank you for joining us for today's webinar. We are going to get started here in just a few minutes, but before we do, I would like to go over a few housekeeping items with those of you that have come into the room. For those of you that have already joined us, the first thing I would like to make mention of is our chat pod, which is still located on the left-hand side of your screen marked chat. Please make sure that if you do have any questions regarding today's content, to go ahead and type them there in the chat pod, and then we will house them and get those answered during our Q&A session that will be held at the very end of today's event and today's workshop. Also, please utilize this chat pod should you experience any technical issues. We do have quite a few people that are watching the chat pod today, looking out for technical issues. So if you do experience any, please make sure to let us know there in the chat pod, and we will be sure to get you taken care of. All of your audio for today's event is going to be streaming through your computer speakers. So if you do have any issues with the audio, please make sure again to let us know there in the chat pod, and we will make sure to get you taken care of. This is our first event on our new platform. We do appreciate everyone's patience in working with us through getting registered and getting into today's event. We do know some of you experienced some technical issues, and we are currently still working with some others to make sure they are in the event as well. So we will have a customer satisfaction survey at the end of today's event. If you did have any bumps along the road, please make sure to let us know, and that way we can address those and hopefully provide a better experience in the near future while we are delivering our webinars and our live events. It is the top of the hour, so I'm going to go ahead and mute my line, and then we will get started here in just a couple of moments. Thank you for joining us today. Good afternoon, everyone. My name is Josh Vickers, and I am with RTI International's Forensic Technology Center of Excellence. Today, we are kicking off our workshop series, Probabilistic Genotyping of Evidentiary DNA Typing Results. And today is our very first module, the elements of DNA profile interpretation of probabilistic genotyping. We do have 3 presenters today. I'm going to introduce our very first. This is Dr. Tamara Moretti. She is a research biologist for the DNA support unit of the Federal Bureau of Investigation Laboratory. She earned her PhD in genetics from the George Washington University for her research at the National Institutes of Health. 23 years ago, Dr. Moretti joined the FBI Forensic Science Research Unit, where she performed research, development, and validation of PCR-based methods for the analysis of nuclear and mitochondrial DNA. She has lectured extensively and trained forensic scientists and attorneys throughout the U.S. and internationally. Just a quick disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without any further ado, I would like to hand the microphone over to Dr. Moretti to introduce our other 2 presenters and speakers of the day. Dr. Moretti?
Thank you, Josh. I'd like to welcome you all as well, and thank you all for joining us today. I hope we succeeded in reaching out to members of both our forensic DNA testing community as well as the legal aspect of our community, as we are trying to share information, our knowledge and experience that in probabilistic genotyping with all the folks, both in laboratories and the courtrooms, who are dealing with probabilistic genotyping evidence. As we transition as a community from our traditional, historical, or what we refer to as the binary methods of DNA profile interpretation and statistical analysis, we have looked for a way to share the information that we have. So we're really grateful to Gerald Laporte and his staff at the National Institutes of Justice, as well as RTI International and the folks at the Forensic Technology Center of Excellence for supporting our ideas and plans to get this information out to you. And especially grateful we are to Josh Vickers for leading our effort here with this webinar. Everything that you have seen on the internet and seeing today are the efforts of Josh and his staff. So thank you, Josh. I want to spend a moment before I introduce the speakers telling you a little bit about our series. Our intention here is to help educate both laboratory scientists and managers, as well as, say, attorneys and judges, in this form of probabilistic genotyping that so many in the U.S. and other places are transitioning to for interpreting evidence, DNA evidence, and calculating match statistics. We're going to start out with today's lecture talking a little bit about past methods and issues involving mixture interpretation, and then introducing probabilistic genotyping. In our next lecture, we're going to talk about, which will be a week from today, we're going to be talking about the statistical aspects of probabilistic genotyping. And then the next, a week after that, we're going to be seeing 3 different software programs. And we have limited this installment to continuous systems, and we've picked Euro 4 mix, True Allele, and Star mix as ones that we wanted to share with you, and the developers of those programs will be sharing information about their programs, and Mike Coble is going to do a little bit of a comparison of the different programs as well. We're then, the week after that, going to speak about validation of these software systems, and then delve into uncertainty and limitations. And then we're going to have a webinar on representing the statistical weight to our stakeholders and the court, and then have an overview of what is happening in the U.S. legal community with likelihood ratios and probabilistic genotyping. We'll hear from some experts who have testified in admissibility hearings, as well as cases. Now, always in these webinars, it seems that we get a lot of questions. Some of them we know are going to be addressed in a future lecture or a future webinar, and we never have an opportunity to get through all of them. So we're going to hold on to any questions and perhaps organize them into additional lectures, or if nothing else, provide a question and answer period as a final installment to our series. So moving on to today's webinar, I would like to first introduce the speaker after me, who is Dr. Peter Gill. I had recently had the privilege of working with Dr. Gill on a SWGDAM committee for developing recommendations for communicating likelihood ratios. Dr. Gill joined the UK Forensic Science Service in 1982 and began research in DNA in 1985, where he collaborated with Sir Alec Jeffreys of Leicester University. In the same year, they published the very first demonstration of the forensic application of DNA profiling. After his distinguished career with the FSS, Dr. Gill joined, since 2011, has been a professor of forensic genetics at the University of Oslo Hospital in Norway. He led the team that developed the first multiplex DNA system specifically to be used in the UK National DNA Database, which was adopted throughout Europe and beyond. He has specialized in the development of interpretation methods, particularly in relation to the analysis of complex mixtures. Dr. Gill is chair of the IFFG DNA Commission, past co-chair of the MC Steering Group on Methods, Analysis, Interpretation, and winner of the 2013 Scientific Prize of the IFFG. So we're grateful for his participation today. We are also pleased to be joined by, getting my screen to work, let's see. Apologies, little technical difficulty. There we go, thank you. We're also pleased to be joined by Lynn Garcia. Lynn had joined the Texas Forensic Science Commission as General Counsel in 2010. She assists the Commission with investigations. She provides legal advice and tracks development and legislation relevant to the FSC's mission, and ensures compliance with Open Meetings and Public Information Act. She represents the Commission at various conferences and stakeholder meetings. She has over 18 years of experience conducting investigations in response to criminal and civil actions, and in developing legal compliance and trading programs, also advising clients in litigation. She is a graduate of the University of Massachusetts Amherst with honors, and she obtained her Juris Doctor from Georgetown University Law Center in Washington, D.C. in 2000. So thank you, Ms. Garcia, for joining us as well. My lecture today is going to be on forensic DNA testing. I'll speak a little about the origins of CPI and then some of the challenges of mixture interpretation. I, too, have the disclaimer that the names of commercial manufacturers are provided for identification purposes only, and inclusion does not imply endorsement by the FBI of the manufacturer, its products and services. The views that I express do not necessarily reflect the official policy or position of the FBI or the U.S. government. So an overview to my lecture, I'm going to take a look back at why we have used the combined probability of exclusion as a statistical approach. I'm going to give a very basic review of DNA structure as it relates to STRs and our testing process. Today, I hope we are joined by some folks from our legal community or other non-scientists. So I would like us all, after today's material, to really have a good foundation to continue on in our series and understand the terms that we're using and things related to probabilistic genotyping. I'm going to lead you through an electropharogram and describe how to read it, and then we'll delve into some of the specific aspects as they relate to the interpretation of DNA profiles. And I'll conclude with introducing probabilistic genotyping and with a slide as well on what it can do for you. So we as a forensic community here in the United States have 202 NDIS sites. That is for the National DNA Index System. You can see in the second column of the chart here the totals for in the various indices of the National Index System for forensic profiles. And then on the right side of the column describes our annual totals for 2018. 11,000 mixtures, and I'll point out that these are actually the quote-unquote good mixtures, the ones that are suitable for searching at the national level of the database, and partial profiles, over 18,000. We know in our laboratories that the ones that don't get uploaded to the national database are there are quite a lot of that. So this is the work that we are doing, and who we are is on average 11 DNA analysts analyzing and reporting these results per laboratory. About a third of us have what I would say is perhaps a medium or large laboratory with more than 10 DNA analysts, and 2/3 of our laboratories have 10 or fewer DNA analysts. So it's a lot of work that we are processing. In terms of the origin of usage of the combined probability of inclusion in the United States, it was the DNA Identification Act of 1994 that authorized the FBI director to appoint a DNA advisory board. We refer to that as the DAB, and it sat from 1995 to 2000. It was comprised of scientists from state, local, and private laboratories, as well as molecular and population geneticists who aren't affiliated with a forensic laboratory. There was also a representative of NIST, and all were appointed by the National Academy of Sciences or professional societies. One of the areas that was included in the charter of the DAB was to examine statistical and population genetic issues. So a subcommittee was formed to make recommendations to the DAB on these matters. And the subcommittee was comprised of Drs. Frederick Bieber, Bruce Bajoli, Ranajit Chakraborty, and Bernie Devlin. Those are people that we're very familiar with as leading experts in these areas and this area in our field. And one of the publications from the DNA Advisory Board noted that when the contributors to a DNA mixture cannot be distinguished, two calculations convey the probative value of the evidence. The first is the probability of exclusion, and for our purposes here, we could also consider the probability of inclusion. Knowledge of the accused and or victim profiles is not used or needed with this calculation, and it's particularly useful in complex mixtures because it requires no assumptions about the identity, the genotypes of any individual, or the number of contributors to a mixture. Now, I remember many years ago when I was in training as a forensics examiner, this was one of the things that was underscored for us, that we didn't have to delineate an exact number of contributors to a mixture, and we didn't have to have a reference genotype, a reference profile for any individual in order to calculate a CPI. The probabilities derived are valid and for all practical purposes were at that time considered conservative. However, the PE does not make use of all of the available genetic data. The second calculation that they supported was the likelihood ratio, and it does consider the identity or the genotype an actual number of contributors of the observed DNA mixture. Certainly, the likelihood ratio makes better use of the available genetic data than does the probability of exclusion. The DIB finds that one or both PE or LR calculations are acceptable and strongly recommends that one or both calculations be carried out whenever feasible and a mixture is indicated. Now, David Kay, who is a distinguished scholar at Penn State Law, in one of his papers, I'd like to share some information with you. In the People versus Simpson case, Professor Bruce Weir suggested in a report prepared for the state that the proper analysis of mixed stains required likelihood ratios. The defense objected vehemently, telling the court that there was no precedent for using likelihood ratios in criminal cases and that we know of no precedent for the admission of likelihood ratios in a criminal matter. Now, apparently, Dr. Kay cites a number of cases where that the contrary would be true, but the defense argued at that time that likelihood ratios are difficult to understand and likely to confuse a jury. So this is where we stood in the 1990s, and that was around the time that research was beginning in STRs. We at the FBI laboratory went online with STRs in 1999 and used the CPI calculation. NRC 2 report in 1992 suggests, though, that likelihood ratio is particularly appropriate when evaluating mixed stains. So how have we in the U.S. been calculating evidentiary weight for mixtures? Largely, we have been using a random match probability. That, for example, would be suitable if you can distinguish a major contributor to a mixture, or a CPI or CPE. And likelihood ratio as well has been calculated by a smaller number of laboratories. And this approach is what is manually done by the DNA analyst, and we refer to that as a binary approach because either or, yes, no, above, below type of decisions need to be made in this process of profile interpretation. But based on this 2018 survey taken by the CODIS unit at the FBI laboratory, which included just about 200 reps from Endus Laboratories, or the TL survey, we have about twice as many laboratories today using probabilistic genotyping than CPI. And this year in 2019, one second, I'm not moving forward again. Sorry. In 2019, we have a large number of laboratories that are in their planning phases of acquiring or validating or implementing probabilistic genotyping system. So this was the idea, the origin of this webinar, that we have so many people using a variety of different approaches at this time, and they are in the process of transitioning to probabilistic genotyping. When it comes to our binary methods, we are trained, we are guided by groups such as SWGDAM that has published recommendations and guidance documents. We're very practiced. Our procedures and us as analysts have matured. We're very conversant with these statistics and very comfortable and have expertise. So we are attempting in this process to help this transition to probabilistic genotyping. Just very briefly, the steps in nuclear DNA testing of evidence. It may be preceded by body fluid testing, but then the first step in the process would be to collect the biological material and extract the DNA. The extraction process is either what's called differential, that can separate sperm and non-sperm DNA into separate samples, or it's non-differential, that just generates one DNA extract. We then, in accordance with the quality assurance standards for forensic DNA testing laboratories, quantitate the DNA because we will be putting a measured amount into the PCR. We don't want to unnecessarily consume the evidence DNA by using too much, and we don't want to use too little and risk not being able to get the typing results. The products of the PCR are then analyzed through a capillary electrophoresis, and then we interpret those results, compare them to the DNA profiles of known individuals, such as a suspect or victim in a case, and then we may perform a statistical analysis to convey the weight of the evidence. Very briefly, DNA is contained within the cells that make up our body tissues. It's also present in the cells of our body fluids, and it is condensed and highly supercoiled into structures that you can see under a microscope called chromosomes. And if you were to unwind those, you would find, if you could see it, the DNA helix. The DNA helix is a twisted ladder with the rungs where A base pairs with T and G with C. So how this relates to our discussion today, we're going to look at just one of those strands. As you can see from what I'm depicting, we have a segment, AATG, which is repeated 4 times. This is referred to as short tandem repeat. These are the locations that we test in forensics, and most of the locations of TR test are these 4 base repeat units, which are called tetranucleotides. On the left here, we have what's called a karyotype. It's all the chromosomes that would be in a male individual's DNA. They're organized by pairs where one set of chromosomes is contributed from the father and one from the mother at the point of conception. So this STR locus on the right with its 4 repeats is coming from the paternal chromosome. We also, from the maternal chromosome, have in this example an STR location that has 5 repeats. So the individual's genotype at this location would be 4-5. In the process of testing this DNA, we're going to use the polymerase chain reaction. Now, there are plenty of tutorials and videos on the internet if you'd like more information about PCR. I won't go into it here, except to say that it allows us to target STR and other regions in the DNA so that we can take sub-analytical levels or minute levels of DNA that we can't characterize in any way except to make copies of them so that we can detect them. Note here that this is just one single locus, and there's a size difference in terms of the length of the amplified fragments for allele 4 on the left and allele 5 on the right. There are actually 20 loci, 20 STR loci that are listed there. Starred are the original 13 that were the core CODIS loci dating back to 1998. But in 2017, we implemented 20 core CODIS loci that are used throughout the United States by labs that participate in the National DNA Index System. So these profiles that are in our national database have typing results from the same loci and can be compared all throughout the country. In this PCR process, we use kits, and they're available from various manufacturers, and they enable us to amplify all of the locations shown here, as well as some others, called multiplexing in a single tube. Now, our process incorporates a fluorescent tag or dye into each of the fragments, and that, after we separate out these different fragments through the process of capillary electrophoresis, is going to allow us to detect and distinguish those fragments, both in terms of their size and color. So this is just a little part of a capillary electrophoresis instrument shown where we have a sample tube, and simultaneously samples are being injected into these hollow tubes, which are called capillaries, and there's a sieving medium inside of these capillaries. So a DNA fragment that's very small has an easy time sneaking through this sieve to get to the other side, and it's going to come out faster than a long DNA molecule that has a harder time working its way through the sieve. So in this process of CE, we separate out fragments of their various sizes. And when they go past this detection window on the far side, there's a laser that is aimed at the fragments, and it causes these dyes to emit light of a various wavelength that corresponds to a color. The computer associated with this instrument is going to collect those emissions of light and also the intensity so that we can correlate the wavelength or color with the degree of the signal relative to a period of time, which correlates to the size of the DNA molecule that has been amplified. So all of that at the end of this process is depicted in a result such as shown on the screen, an electropherogram. I know you can't see any of the detail here, but I wanted to note that we have, with the green bars, we have different locations in the DNA represented. On top are the blue labeled loci, followed by green, and then yellow, which is depicted as black, red, and purple. This is just one of the kits that is approved for usage at the National DNA Index System for forensic samples, and there are other kits that labs can use as well. I'm going to zoom in on the bottom panel, which is shown in orange, and this is an internal size standard. After the sample has been amplified to the PCR, a small portion is taken into a new tube, and to it is added this internal size standard, which acts as an internal ruler in each sample. And what it does is we know the size of that particular peak. It's 80 base pairs in length, and that one is 100. So looking across the different panels, this internal size standard to the sample allows us to figure out, or the software associated with the system, to assign a size to all of these fragments. So we have the small ones on the left and the large ones on the right. Another sample that is subjected to capillary electrophoresis for each of these sample sets is called an allelic ladder. That is shown here, and you can see at all the various locations there are many different alleles that an individual can have. So you can imagine an individual having one allele here, or a pair of alleles here, and so forth, all throughout this profile. And it would be exceedingly unlikely for two different individuals to have the same exact genotype of two alleles at all of these various locations. Now, just as the alleles in the sample are sized, the alleles in this allelic ladder are sized. So it's the comparison of the allelic ladder information to the information from the sample that allows these alleles to be designated with a number. And the number is an indication of the number of short tandem repeats in the sample. Here's an example zooming in on the red labeled loci, and they are all heterozygous, which means that there are different alleles. The first one, for example, is an 11 and a 17. And one thing to note is the nice balance of the alleles across the loci. We have on the left, this is on a scale of peak height. It's a measure of that light intensity. So generally, the taller peaks represent more DNA being detected than shorter peaks. So that's how we correlate that information. The boxes indicate the allele number, the length in terms of base pairs of DNA, and then the height in RFUs, which stands for relative fluorescence units. Now, here we have two homozygous loci on the left. So this is a heterozygous 7.9.3, and the peak to the left, homozygous, is actually two copies of the 13 allele. And you'll note that the shared alleles, the 13-13, is approximately in this well-balanced, undegraded sample about double the height of the individual alleles shown to the right. When you see on these allele names, the 9.3, or up on the right, the 21.2 or 25.2, these are partial repeat sequences. So it'll be full repeats and say 1, 2, or 3 extra bases. in the allele. One of the bits of information that is useful to us is calculating a peak height ratio. That is a ratio of the smaller to the taller allele. And we see in the examples here, these are nearly equivalent in terms of their height, and these are a little bit different. So this is just normal variation that we see from the amplification process and sampling of that DNA extract and the amplified product for CE and so forth. Another or similar way to compare the two peaks is through heterozygote balance. That is a little bit more information because it orders the alleles. It's the leftmost allele divided by the height of the allele on the right. So we use this information to help us determine whether two alleles could have originated from the same individual. Here's an assessment from our kit at the FBI laboratory in terms of the peak height ratio of thousands of alleles, thousands of loci. And what we are looking at is the peak height ratio on this scale and then the average peak height. So over here, these would be lower template samples that are amplified, and then more and more DNA template going up to probably a nanogram of DNA. And what we have calculated here is the average peak height ratio, sorry, not yet. The peak height ratios for the individual samples are plotted here. And if we were to mark what we see that is within about a 70% peak height ratio, we see that we're looking at peaks that are above 5,000. And in the range between 2,000 and 5,000, the peaks are generally within about 60% of one another. And even lower than that, we have from our analytical threshold, which is the bottom line of our detection, up to 2,000. The two peaks could be as different as about 50% of one another. So this is information that we use in analyzing or interpreting profiles to help us determine whether peaks could be associated with an individual's genotype. This is an example of degraded DNA, and we recognize that by the fact that the smaller DNA fragments on the left are detected, and then there's a downward slope such that the large fragments or loci on the right did not yield any amplifiable fragments to a detectable level. And it's around the height of about 220 that in this particular sample, we're unable to detect the type. That simply means that the DNA template, which would be, for example, the DNA from the evidence, has been exposed to some conditions. Some may be heat or humidity or exposure to chemicals that cause the DNA to break up, and it's fragmented such that the primers that make the copies of the DNA don't happen to sit down on an intact target of the DNA for amplifying. So in degraded DNA, we generally see the types over on the left side of the profile. Also, on this electropherogram, we see the loci that are useful determining the sex of the donor of the sample. This is the amylogenin locus, and the sample is from a female. So we see only the X chromosome, which would represent the XX genotype at this location of a female. If it were a Y, we would see an extra peak. I'm sorry, if it were a male, we would see an extra peak there representing the Y chromosome and see the XY genotype for a male. Today's kits include additional markers for the Y chromosome, and because this is a female sample, there is no Y typing information shown here. Now, over here on the right, because the sample is degraded, as I know from the other locations, I would not necessarily expect to see a Y typing result for this, even if this were a male sample. These are the various alleles from the allelic ladder showing the possible Y chromosome alleles that could be detected with this particular kit. So now we want to move on to the topics of sensitivity and stochastic effects. Now, we work with evidence, so ideally we want to have a sensitive system that can detect the typing results, so detect the DNA from the evidence, but there is a cost to sensitivity that we're going to be talking about. This is looking at amplifying a nanogram of DNA, which is our target template amount that we use for the PCR. And if we conducted 27 cycles of the PCR, the blue line, versus adding an additional cycle, 28, which is going to make more copies and generate a higher average peak height in red, and then 29 cycles. So this is a standard test that we would perform in validating a system to figure out how many cycles would we be interested in using as part of our standard operating procedures. Let's talk about stochastic effects. This is obviously a dated slide for those of you who know a little bit about football. So this slide was made a few years ago, but the stochastic effect is exemplified by the flipping of a coin. If you flip a coin 2 times, you might get 2 heads. That doesn't mean that you have a 2-headed coin. It simply means that you haven't flipped the coin enough times to see an accurate representation of what is actually on the coin. So when you flip the coin too few times, or in terms of DNA testing, if you don't have enough DNA to be representative of the individual's genotype, you might observe what we see as a stochastic effect. If you were to flip the coin more times or have more copies of the DNA template for the PCR, you would see that you have a better representation of what the sample actually is. And again, they don't necessarily amplify up to a equal level. We can see some imbalance in the peak heights as shown here. So with the stochastic effect, 2 things can happen. We can see an imbalance in peak heights of 2 alleles at a heterozygous locus, or we may see one allele fail to be detected at all, and that is referred to as allele dropout. The original stochastic threshold goes back many, many years, and this, I know some folks in laboratories are not familiar with the system. It's that old, but some of us have worked with the amplotype DQ-A1 or DQ-alpha or DQ or polymarker typing strips. This was one of the first PCR-based methods to follow the original RSLP methods used by forensic laboratories. And they have what's labeled as a C dot, or maybe that stands for control, and the other system, it was the S dot, maybe that stands for stochastic. And this was the lightest dot on the, well, it was designed to be, if a low-level DNA template was present in the sample, this dot would not light up if you were in the so-called stochastic range, where you might be experiencing the loss of allele due to stochastic effects. So it was actually in the 1990s when Bruce Bedoli and I were giving an STR road school in the state of Texas that someone from the audience asked, we've had this stochastic control from our DQ-alpha polymarker system. STRs don't have a way to determine whether you might be having allele dropout. And Bruce answered by saying, well, we can use peak height to serve as a threshold for defining the range within which we might be concerned that we may not have detected all of the typing results at a locus. And we included that in our STR validation paper in 2001. The stochastic threshold is defined as the peak height value above which it's reasonable to assume that at a given locus, allelic dropout of a sister allele has not occurred. In 2010, SWGDAM incorporated this idea of the stochastic threshold in its guidelines for autosomal DNA testing. So back to this issue of sensitivity of detection and how many cycles we want to use. This is showing the 3 different cycle numbers we tested at the FBI laboratory with 125 picograms. We are looking, this is called a heat map, and we're looking at location number 1, 2, 3, and 4, et cetera, and a single typing result here, and then these represent the various samples that were amplified at 27, 28, and then 29 cycles of PCR. Where both heterozygous alleles are detected in the profile, it's shown as green. Where we had dropout of, and by the way, these are all heterozygous alleles shown here, while dropout of a single allele was indicated, was shown, it's highlighted in yellow, and where both alleles were lost, it's highlighted, it's shown as red. So we're going to go down the scale in terms of DNA template amount. We started to see more loss of alleles or failure to detect alleles at 62 picograms and going down further to 31 picograms and 16 picograms. We start to see the inability to detect more and more of those alleles. And it's sort of visible here, I think, as we go from the lower cycle number to the higher cycle number, more red here, less red down here, that more cycles, we have less loss of alleles. So on the one hand, you might think that higher cycles is better, but higher cycles comes with a cost. And by the way, the highest peak of the remaining sister allele, so this would be the height of the allele that was detected when the sister allele was lost throughout our entire data set, which was almost 9,600 heterozygous loci analyzed at 28 cycles, which was a cycle number that we selected. The highest height of the remaining allele was 702 RFU. So we set a stochastic threshold for this system at 725 RFU. Well, in response to the FBI or Bruce Bedoli's mixture paper published in 2009, Peter Gill and John Buckleton very aptly observed that one of the issues with using a stochastic threshold is this falling off the cliff effect. An allele at what was then a stochastic threshold of 150, an allele at 149 RFU is subject to different guidelines than a peak that differs in height by a single RFU, and they are absolutely correct in that observation. And in fact, a peak that is quite different is treated the same as long as they're both under the stochastic threshold. So we have a situation where things are treated differently, even though dropout of a sister allele might be very improbable, but things are treated the same if dropout of a sister allele is different in that way. They commented that probabilistic models would circumvent this requirement for a threshold. We simply needed a software system that could be used by the many laboratories throughout the United States that would help us with this process. I'm now going to step through some examples of electropherograms so you can see what the results look like at these low levels. And these are single source profiles. And I have 4 replicates at each of the low template amounts. And so perhaps in this set, let's say maybe focus on the left side here, and we'll see how there's variation in peak height from one PCR to the next. Okay, this is stochastic amplification. This is 125 picograms. And then if we were to drop down to 60 picograms, we see up top with that black triangle the dropout of one allele. A replicate, we have more alleles that happen to not be detected, and so forth. So different replicates, this is exemplification of stochastic effects. Here I didn't bother at 31 RFUs to put a triangle because there's so much below the analytical threshold of 150 RFU. I'm sorry, of 50 RFU, not detected. So you can see some variation there. And even going down to 16 picograms with the system we currently use, we can get some alleles detected, but not many at all, and it's variable from PCR to PCR. So the downside that I mentioned earlier about using a higher number of cycles or enhanced sensitivity is allele drop-in. And this is a lot of information here from our validation study, but it's addressing drop-in in our testing of one of our methods for extraction. So these are samples, these are extraction blanks. There's no biological material put into these samples, and this is another instrument we use for extraction. We have negative amplification, no DNA put into PCR reactions, that's the AMP negatives, and then we typed some actual samples as well. And you can see here the total number of loci. These two columns are added here for our 28 cycle data. We are comparing 27, 28, and 29 cycles in terms of allele drop-in. Here are the tallies, and this is the total number from the different extraction processes or the different blanks or negative controls, the total number of drop-in peaks that were detected. 58 out of 20,000 results in this drop-in rate. And so you can see here along the bottom, a very low drop-in rate if you have fewer cycles, and a higher drop-in rate as you increase the number of cycles. What we observed in short here for the 29 cycle protocol was 62 drop-in peaks on 3 plates of data. And we felt that was really just too much. The 28 and the 29 cycles were both suitable, but we decided to go with this particular cycle number because of its enhanced peak height relative to the other with a very low level of drop-in. And so fortunately, with the probabilistic genotyping systems, we have the ability to model this drop-in. I want to move on now to the topic of stutter. Stutter is an aspect that is inherent to the amplification of short tandem repeat loci. On the top here, we have a template of DNA that's going to be copied with short tandem repeats. And this is the growing DNA strand that is being copied. Now, during this PCR process, which is accomplished through a protein called a DNA polymerase that sits down on the DNA and causes this copying to occur, there's something called processivity, which is limited. It means that this DNA enzyme, this enzyme can only add so many bases to the growing DNA strand before it falls off. But that's okay because another one jumps right on to the growing strand, to the template, and continues that copying process. So it's dynamic in that sense. Well, during that process, though, the growing strand can, we call it breathing, it can become unattached to its template. And when it joins back down to the template to continue the copying process, it can loop out. And if it does so, as shown here in the middle panel, it's going to align simply because it's supposed to be here, but it's aligning one repeat unit farther back. This generates a product that is going to have an extra repeat unit compared to what it should have. And that's what we refer to as forward stutter. And then if it is the template strand that loops out and the misalignment occurs, we're going to miss copying this particular repeat unit, and the amplified product is going to be one repeat unit short, and that is referred to as back stutter. Those are shown here on the electropharogram, back stutter in black, where the first one is a 16 allele with a back stutter missing a single repeat, so it's detected as a low-level 15 peak, and then the forward stutter peak, which has an extra repeat unit, and it is thus a 17. You'll notice that relative to the source peak of this stutter, these are very low-level peaks. But you can imagine that if you had a mixture of DNA where you had a major contributor who contributed more DNA than, say, a minor contributor that had lower peak heights, or a trace contributor, that we would then have a contributor to the DNA that was in the same height range as these stutter peaks. So this is how the presence of stutter can complicate the interpretation of mixtures. But one good thing is that it's fairly reproducible, the degree to which the stuttering occurs. And we can characterize the different loci for the extent of stutter that we have seen through our controlled studies. This is an example of a different type of artifact. It's one that occurs at SE33, that locus, and the D1S1656 locus. And we see the actual stutter peak here is an 18, and then the allele 19, and it's this intervening peak here that is 2 bases shorter than the source peak. These are our guidelines for manual interpretation of DNA profiles. So you see the various loci and our maximum expected percentage of stutter for this locus D3, it's called in terms of shorthand. It has 11% stutter expected, which is the average that we saw in our studies plus 3 standard deviations. That is the back stutter of 1 repeat unit. The forward stutter of 1 repeat unit is 6%. The forward stutter is always seems to be less than the reverse stutter, the back stutter. And then at those 2 loci I showed in the previous slide, we have expectations for the maximum stutter for the 2 base position. This, though, that is how we have interpreted stutter peaks historically in our STR data. This is how we interpret stutter in our software for probabilistic genotyping, and it's calculated as a stutter ratio, which isn't a percentage; it's a number as shown here, and it is assessed on a per allele basis. So this is the D3 locus, where we saw in the last slide that the manual guideline is 11%. So we have allele 13, 14, and 15, and so on. And stutter increases. These are the percents of stutter that we saw in a study for allele number 14, and then the instances of stutter for allele 15, and so forth. And the line of best fit for all of these data points is shown here dashed. With allele 14, the expectation is that it would be around 5.3%, allele 15, 6%, and so on throughout the data. And what's interesting to note, and what is the advantage of being able to use software with its greater complexity to do all of these various, the complexity of all these calculations and such in single source and mixed profiles, is that the manual guideline, the average plus 3 standard deviations taken as a locus as a whole, is way up here at 11%. So for example, if we were to see in a case sample an allele 14 that had a 9% peak in the 13 position, then we would know that that is very unlikely that that is a stutter peak, because allele 14 doesn't stutter up to this degree of 9%. So that's one of the ways that probabilistic genotyping, or using a computer for this assessment, is advantageous to us. These are the forward stutter ratios, and all of the loci here have a manual guideline of 6%. I'm showing here in red, just so you can see it, what the scale caps out at. So this is 4%, and all of the data here for forward stutter at this SE33 locus is under 4%. Well, up here would be the 6% that we are using for our manual interpretation. So again, advantage of using software through probabilistic genotyping. We have challenges, as I mentioned, when the sample has similar peak heights to the stutter peak. So a peak in the stutter position, it can originate from stutter. It can be allele, or it can be a combination of both stutter and allele shared at that particular position. It's difficult to manually interpret a minor contributor allele in the stutter position of a major contributor allele when the minor contributor and stutters are similar height. Dr. Badoli suggested that if a peak under the stutter threshold is determined to be allelic and is used for matching and statistical purposes, then the stutter could be removed from that locus. Well, it would have been removed if you're considering it an allelic peak. But if you're going to do that, then you should remove the stutter filter from all the other loci. So one question we have to ask ourselves is, how could you look at any of those low-level peaks that are underneath your stutter expectations and actually determine that it is an allelic peak? And there really isn't a value to unlifting the stutter filter, because that would essentially disqualify so many of those loci in terms of the stochastic threshold, or if usable, our statistic would be hugely diluted unnecessarily. So this is just the dilemma that is presented when we have stutter peaks and we have low-level contributors, and how we have used this approach to disqualify a peak as a stutter peak if it meets those stutter thresholds in our manual interpretation process. Very quickly about spectral artifacts and the analytical threshold. Now, as we talked about the CE process and the amplified fragments are giving off their fluorescent light, it fluoresces along at various areas of the spectrum shown here, the various wavelengths. And you can see, for example, that this blue spectra overlaps with the green and also some of the others. So in the process of analyzing the data, the computer associated with our instruments separates out these spectra. And but what we can see sometimes is what I have historically referred to as bleed through or pull up. Now, bleed through, as I've used the term, has been for when the source peak is on scale. It is not exceeding the maximum detection capacity of your CE camera. So here is a peak in red, and we are seeing that it is actually bleeding through so that a peak is detected in blue. And it doesn't necessarily this is not adjacent colors. It goes blue, green, yellow, red. And then down here in purple as well, we see this peak bleeding over as well. And note that the one coming from the red is actually labeled as a 12. So and pull-up would be the situation when these peaks are off scale. Off scale on the instrument that we use is around 30,000 RFU. And so these ones are on scale. But the challenge would be that if you have really tall peaks like this, that sometimes a pull-up or a bleed-through peak is going to fall within a bin. And it might be consistent with bleed-through, but you could not discount the possibility that this could be an actual allele of a minor contributor. So this is how complex samples, we have to make determinations. It might be appropriate in such instances to re-inject the sample for less, but so that these bleed-through situations are not detected, but in so doing, you might lose minor or trace contributors to your profile. Replicate injections of the same sample, sometimes these artifacts will appear and sometimes they would not. So that's another approach to eliminating these spectral artifacts. And they might be labeled by the software as spikes or off-ladders or alleles. Now, in terms of the analytical threshold, there are a few different ways that could be determined. Typically, we might look at regions of the electropherogram and also in extraction blanks where there are no peaks, and we'll just measure the height of the signal along the electropherogram. What we found in the validation of our most recent kit was we looked at using different analytical thresholds for the same data set for testing. And if we had an analytical threshold of 50 RFU, we had an enormous number of non-allelic peak edits, even if we were using a global filter to remove peaks at 2% of another peak in the electropherogram. This is 486 edits to 106 1 nanogram samples. So this is obviously not operationally usable. So we tried testing a higher and higher and higher analytical threshold. Now, note that with our ProbGen software, we have to analyze with no global filter. So even an analytical threshold, which is this minimum detection level that we set in the software, of 100 RFU would really still have too many edits that would be required for essentially a plate's worth, or a little more than a plate's worth of sample. So we, for this reason, have an analytical threshold of 150 RFU. I want to move now on to DNA mixtures. A mixture is defined based on analyzing the entire DNA profile. It's typically defined as 3 or more alleles that are present at 1 or more loci, although rarely there can be something called a triallelic locus, which is an individual who at one locus has, rather than 2 alleles of maternal and paternal origin, they have an extra allele because the target that we're amplifying in the DNA is present in an extra location. It's been replicated at some point. The peak height ratios between a pair of allelic peaks for 1 or more loci are well below those expectations, the 50, 60, and 70%, because those peaks are coming from different individuals who are contributing perhaps different amounts of DNA. That would cause the height of different peaks at a locus to be outside of our expectations for if 2 peaks could go with a single individual. So as part of the process of interpreting mixtures, we estimate the minimum number of contributors, and this is generally done by selecting a locus that exhibits the greatest number of allelic peaks, but also we take into consider the relative peak heights, which could be indicative of different individuals. We know from experience that in our kits, this is a mixture, I believe, of 3 individuals. That's a 1-to-1-to-1 mixture, 3 people with the same amount of DNA totaling a nanogram of DNA. And this is what this mixture looks like. And these loci that I boxed in, just in my experience, happen to be the ones that there's a lot of variability, meaning that different people have different alleles. You'll see here at this location, which is D22, these 3 people they share a lot of the alleles. They only have 3 different alleles, but they have many more alleles down here, useful loci. Now, peaks that are shared, whether we're talking about 2 alleles from different individuals that are the same, so person 1 has a 13 and person 2 has a 13, or whether there is sharing of an allele with a peak in the stutter position, the greater the number of contributors, there's a greater potential for peak sharing. Peak sharing, because you have no way of knowing what the height is of the constituent peaks, peak sharing reduces the value of using peak heights to help you deconvolute a mixture. Shared alleles with low RFUs can be masked by peaks with higher RFUs. In fact, if the difference in height is so great, we may not even throw the peak height ratio of that major contributor off enough for you to even recognize that an allele is being masked through the sharing. And sharing can also artificially place an allele that's below your stochastic levels. Sharing can cause that stochastic peak to exceed the stochastic threshold or your stochastic expectations. So all of these topics that I've talked about, number of contributors and allele sharing of atypical or expected types of stutter, the peak height variance, especially within a locus, allele drop out, allele drop in, the spectral artifacts, DNA degradation, especially if your evidence has DNA or biological material that was deposited onto the evidence at different times under different conditions, where the previously deposited material might have degraded, but then subsequently biological material is deposited on the evidence. You extract both of those sources. One is degraded and one is not. I call that differential degradation among the contributors. And the rare triallele are just some of the things that contribute to complexity and the typing of mixtures, the interpretation of mixtures. But we have to ask ourselves the question, is this too complex for me? Well, some of them may be, as the human, factoring all of these different things in, but is it too complex for a computer to do a probabilistic genotyping software? I've heard many people talking about their procedures that they use in their laboratories, and they might say, well, we don't even touch profiles that have this feature or that feature or lack this or that. Well, I would just encourage folks, rather than taking our experience and knowledge and expectations that we have from all of our years of experience as humans interpreting mixtures, rather than use that experience to influence what we will and won't do in the ProbGen world, why don't we just test all of those types of profiles and the data through our probabilistic system using known samples that we know who's in what proportions and what mixtures, and see what the ability is of our software, to the ProbGen software, to handle these more complex situations. Because certainly, as all of these things are sort of shown collectively here, stacking or sharing of two alleles that are truly at stochastic levels, and they are above stochastic expectations here, or accentuation of peak height of an allele due to sharing with stutter. We have an allele here that is just discounted as stutter. Over here, we might have DNA degradation or a dropout of a locus. Down here, we have a masked allele with some stutter and a dropped allele. Both of these here at this locus are below the stochastic threshold. Both of them here are accentuated by sharing with stutter peaks. Then we have, and so forth. All of these types of situations are things that can add to the complexity of me interpreting the results and deciding what to do with these low-level peaks, but can certainly be handled by a well-designed and validated probabilistic genotyping software. So we have the situation, though, that we have different analysts following the same protocol getting different results. And we've seen this through the Mix13 study that was conducted by NIST. And then we have experts that use different protocols. And one, for example, might calculate a CPI using a binomial approach and wasn't able to use all of the genotyping information because the loci were disqualified for falling below the stochastic threshold. And then we have another expert looking at the exact same data. They calculate an LR using a probabilistic approach, using all of the genotyping information. They've assumed it's a two-person mixture. They can assume perhaps that the complainant was one of the contributors. So we have a situation where the same evidence could potentially, in some situations, say, be presented in court, and this has occurred with different approaches being used and different numbers being generated. And this is exactly what we would expect, given that the different approaches are different, but it certainly would be nice if we were all moving, and which we are, to the same general aspect of typing. Because these safeguards, the stochastic threshold that we've put into place to avoid the potential of a miscall, has cost us the loss of information, and also by disqualifying peaks in the stutter position, it as well has caused the loss of information. And in avoiding the potential for the miscall, what we have done is reduced our statistical power. The match statistic would be more common. This is a slide courtesy of John Buckleton and Joanne Bright of ESR, and we see here the log of the likelihood ratio. Basically, the line shown in the middle is a LR of 1 or a log LR of 0, and these would be your inclusionary results and your exclusionary results. And you can see here for the data that they tested for very, very low average, very low template amount data, that we get very poor data because there's not a lot that can be used in the analysis, but the probabilistic analysis that allows us to use more of the typing information generates good data for likelihood ratios. So what is probabilistic genotyping? It refers to the use of software for biological modeling. That, for example, is a way that you tell a computer how to deal with a biological phenomenon like stutter. It uses statistical theory and computer algorithms, as well as probability distributions. Probability distributions example would be, what is the probability of dropout if my peak is 100 RFU or 500 RFU and so forth. And this software is going to infer genotypes from single source and mixed DNA typing results and calculate a likelihood ratio that expresses the weight of the evidence. Now, probabilistic genotyping software still requires a human. Most of them, as I understand it, require, well, you certainly have to review the profiles and determine and interpret some of the data as artifacts. Whatever can and can't be accomplished by the different software has to be addressed prior to usage of the ProbGen software. And most of them, as I understand it, require some decision as to a number of contributors for the samples. And then, just because the probabilistic software has analyzed the data, done a comparison, and generated a statistic, that is not necessarily the bottom line. That you are still the expert that has to review the input data and the output data, because the probabilistic software is not an expert system. Expert system, in terms of the ENDIS procedures, has a very specific definition and is not at present approved for casework. It's only at present used for databasing laboratories. Advantages of probabilistic genotyping: it narrows the range of potential solutions to the deconvolution of a mixture by providing the ability to use more genotyping information. There are no thresholds. And it accounts for uncertainty by incorporating variance parameters and probabilities of allele dropout and drop-in. It can remove some of the subjectivity in the interpretation of DNA typing results. And this sounds sort of like a marketing statement, but it's my statement, and it really is true, that compared to the traditional methods used in the U.S., it's a more powerful tool for supporting the inclusion of true contributors and the exclusion of false contributors based on all of our validation studies. I want to go through just a kind of a fun example of the concept of what is happening in some of the software programs with something called the Monte Carlo Markov chain, or MCMC. So many of us have had this experience of sitting before this machine as part of a vision test, and the optometrist looking at you has no idea what prescription you would need. And so he takes a shot in the dark, and this may be the result, that you can see this. He flips those little dials and puts another lens in place and asks you, which is better, 1 or 2? And you would say 2. He then makes another choice and says, which is better, 1 or 2? And you would say 1. He then asks you again, and you do this many, many times until you're satisfied that we have a good result for your vision. So a very similar thing, sort of similar thing, is happening with probabilistic genotyping, and that is we have this result as our profile. And the software might propose, well, here's a guess. We don't like to use the word guess, but it's we're going to go through this process of proposing a number of different possible genotype solutions to this mixture and then see which is the best fit, just like we're testing out different lenses in the eye test. So this is a 7-9 contributor and an 8-10 contributor. And the proposal is this very low template and they're about equal proportions. Well, this is a very poor fit for the data. So when asked, is this a better fit or is this a better fit? Well, you would say, well, this is a better fit. It's kind of spot on for some of those peaks there. So I would go with this rather than the first proposal. All right, is this better or is that better? And you might say, well, it looks good for the 2 peaks on the left, but not so much for the peaks on the right. But this is overall a better answer for who the contributors to this particular mixture could be. So imagine this happening millions of times, millions of guesses being made. And after all of this, we arrive at a set of genotypes that explains the observed data well. And that is what probabilistic genotyping is all about. So in terms of the likelihood ratio, what we have typically is a complainant or a victim and a person of interest, which say is the suspect in the case. And the question before the court is this person of interest a contributor to the mixture? Well, on the one hand, the prosecution would say, yes, that person of interest is a contributor. And so the proposition would be that the DNA is from the person of interest. Well, the defense would say that no, the suspect here is not a contributor to the mixture. The DNA is from an unknown individual. The likelihood ratio is merely comparing those two probabilities. The probability of the DNA, given that the results are from the person of interest, and the probability of this DNA result, given that the DNA is from an unknown individual. We can test a variety of different propositions. We might as well need to assume the presence of DNA. Let's say if this were a body swab, we could assume the presence of the DNA from whom the sample was taken. And when the likelihood ratio is 0, now that is somewhat of an artifact of the software, which will truncate an LR that has so many zeros in it. But when it is comes out as 0 or at some threshold determined to be 0, it means there's no support for the prosecution hypothesis or the numerator, and that would be an exclusion. When we have an LR of 1, just looking at this ratio, this formula, it means that the two hypotheses are the same. There's no greater support for either proposition. And this is what Sugdown would refer to as uninformative. An LR greater than 1 means we have greater support for the probability of the DNA results given that they come from the person of interest. And then if the denominator is greater, the LR is going to be less than 1, and that would be support for the defense proposition. Just a word on technology. The terms I've used in that example are HP and HD. This is merely the historical vernacular that's been used to represent alternate propositions. Some argue against usage of these terms because they are set by scientists, usually without consultation with the prosecution or the defense. Several propositions may, in fact, be under consideration, and we may not know which one or either is supported by either party at the time of reporting. So these are simply terms for the propositions that are defined by the scientists. We at the FBI laboratory use the terms H1, H2. You could use HP and HA for alternate or whatever. But the point is that the LR gives a good indication of the ability to distinguish two opposing hypotheses. And I think this may be my last slide. And this takes you back to this is the 28 cycle data, that the cycle number that we are using for amplification with our kit and the different showing the allele dropout. And if we look at data at the same template amounts, as I'm going to show you here, even at 62 picograms, we can still reach the maximum LR. And this, by the way, has 27 zeros in it. It's an enormous number. And that is supported by what we looked at earlier, which is we are getting at 62 picograms a lot of complete profiles. So for this single source sample, 62 picograms can generate our maximum capable, our maximum achievable LR. And then as we look lower in terms of the template amount and start to see, as we look at 125 picograms, where we start to see dropout, we will see the LR going lower and lower and even lower still as it correlates perfectly, is my point, with the observations and that from the data. And that is my general takeaway about having validated and used probabilistic genotyping software, that it is something that is very new to us. Using LRs is quite different. And we're still in the process, many of us, of getting our training and getting used to this change to the new system. But it really does fit right in with our expectations with the data. So I'd like to end by thanking you for your attention this morning. And with that, I think I will pass over to Dr. Gill for him to continue our session. Thanks so much.
Okay, thank you very much, Tamara. I'm hoping that everyone can see and hear me. Okay. Yes, thank you for that talk, Tamara. One point that you made in your last slide, I think I'd like to just start on, really. But what you showed was that you can get a full profile from 62 people.
Recording has started.
DNA. Okay, I'm not sure if you heard that, so I'm going to start again. Yes, thank you for the presentation, Tamara, and I'm going to start by picking up on what you said in the last slide. It's clear that we can get full DNA profiles from just 62 picograms of DNA, but 62 picograms is just a handful of cells. It's about 10 cells worth. Of course, what we're dealing with is an extremely sensitive technique. And of course, this has implications in the way that we report evidence. So what I want to do in my talk is really to go through a framework to interpret evidence so that it's fairly reported, not only on behalf of the prosecution, but on behalf of the defence as well. So the presentation of evidence follows a structure, and I'm going to be leaning very heavily on some work which some colleagues did on behalf of the European Network of Forensic Science Institutes to actually compile this into a booklet. And the interaction between the scientists and the court must certainly follow strict guidelines, otherwise there are risks of miscarriages of justice. And although the framework that I'm referring to here is referring to DNA all the time, because this is what we do, it does actually apply to all kinds of scientific evidence. So this is the booklet I mentioned. It's freely available on the internet, and it can be downloaded at this website. Again, picking up from what Tamara said, we deal with propositions, and when we're dealing with propositions, we are dealing with the likelihood ratio framework. So let's just go through an example. A DNA profile is recovered from a crime scene, and the prosecution and the defence propose alternative propositions. The prosecution would say that the DNA came from Mr. Smith, and the defence would say that the DNA came from an unknown person. And what the likelihood ratio does is it measures the strength of the evidence if a proposition is true. And you'll note that I've highlighted the word if, because this word is the conditional and it is extremely important. So the likelihood ratio is simply the ratio of these two probabilities. It's the probability of the evidence if H1 is true, and the probability of the evidence if H2 is true. So it follows from that if the likelihood ratio is greater than 1, then this means that the evidence supports the prosecution proposition. If the LR equals 1, then the evidence is neutral. It doesn't support either of the hypotheses. But if the likelihood ratio is less than 1, then the evidence supports the defence hypothesis, proposition 2. Now, people often think that when we get a DNA profile, the likelihood ratio value is usually overwhelming, and people often think that the likelihood ratio can only be calculated when two profiles match perfectly. But the important point to make about the likelihood ratio formula is that it can be calculated when two profiles do not match. So what happens is that the likelihood ratio is less than 1. It's a very small number. And we don't have to describe this as an inconclusive or an exclusion. We can actually give a strength of the evidence that favours the defence hypothesis. So in that respect, the likelihood ratio is unbiased. Importantly, the likelihood ratio does not tell us if a hypothesis is true or not. This is not what the scientist is doing. The scientist cannot tell you if a hypothesis is true or not. We evaluate the strength of evidence in support of a given proposition. Likelihood ratios are extremely flexible, however, and they reflect the alternative views of the prosecution and the defence, respectively. And for the scientists to produce these propositions, it's important that these discussions with the prosecution and the defence occur so that we understand the case circumstances. The wording that we use is extremely important. It must be concise and it must explain the points of view that are contended. So it must explain the differences of the points of view between the prosecution and the defence. So supposing that we've got a DNA mixture of a victim and a suspect, the prosecution would say the DNA has come from the suspect and the victim. And the defence would say the DNA has come from an unknown individual unrelated to the suspect and the victim. And what we're doing here is we're conditioning on the victim, and this would be a typical kind of statement if, for example, a vaginal swab is taken, because it's a given that you would expect to find DNA from the victim on that swab. Now, when the scientist goes to court, then he would use this kind of wording. I've considered two alternatives. Mr. X, who is the suspect, and Miss V are contributors to the evidence. And the alternative proposition is that Miss V and an unknown person unrelated to Miss X are contributors to the evidence. And of course, being concise, we have to use the word unrelated because we're not assuming that the unknown individual is a brother, for example, to Mr. X, which would result in a completely different calculation. And the conclusion would be something like the evidence is 1,000,000 times more likely if the first proposition is true rather than if the second proposition is true. So you see that I've used this word if again. I'm not making any statement about whether the proposition is true. I'm only making a statement with regards to if the proposition is true. And in this particular case, we could include a verbal statement to the effect that the evidence provides extremely strong support for the proposition that Mr. X contributed to the crime stain. It is very important to point out a pitfall with all of this. The scientist certainly must not say anything like it is more likely that the DNA came from Mr. X, because this is known as the prosecutor's fallacy. And certainly when this has been carried out in the UK, for example, it has resulted in mistrial. So judges in the UK are certainly very well aware of this fallacy. So what you have to say is the evidence is more likely if the DNA came from Mr. X. We're not saying that it came from Mr. X. it is more likely if it came from Mr. X. So this wording gives the probability of the proposition given the evidence, not the probability of the evidence given the proposition. So to summarise that part, the scientist does not evaluate propositions. We ask, what is the probability of the evidence given the proposition? Now, ideally, propositions are provided by the mandating authorities. So this would be from the prosecution and the defence. And ideally, these propositions should be set before the results are known to prevent bias. So what that means is if you have a case, if the prosecution proposition is true, then you should have an idea what you expect to find if that was the case. And alternatively, if the defence proposition is true, you should have an expectation of what to find if the defence hypothesis is correct. So it's important to think about setting propositions before the results are known. Of course, when you run the test and you find the results, then you may have to modify your propositions as a result of your findings. But it is important to start the process off before you do the testing. But I want to talk now more about the issues which go beyond the DNA profile. And as we've seen, the technique is extremely sensitive. We can detect DNA from a handful of cells. And this inevitably leads to various questions. And in fact, when we go to court, it's the origin of the DNA profile is not the issue. It often is not the issue. It's what the court is really interested in is how, why, or when did the DNA profile become evidential. And in particular, somehow we have to get from the presence of the DNA profile, the knowledge of the DNA profile, its association with an individual, and then we have to somehow associate that with the activity that led to the transfer of the DNA profile so that it became evidential material. So the sort of questions that would be asked will be things like, well, was it direct at the time of the crime or was it an indirect? Could it have been an innocent transfer? And these are the sort of discussions that we increasingly see happening in court. So what does a DNA profile do? Well, we know we can identify individuals, but what doesn't it do? Well, the mere presence of a DNA profile with no other information, it doesn't tell us anything about the how, why, or when it became evidence. So now I want to discuss what we can do about this, and I want to describe the hierarchy of propositions framework, which is a framework which was developed by Ian Evert and his colleagues at the Forensic Science Service. Okay, I'll start at the top level. The court is interested in the ultimate issue. We call this the offence level. And the court asks the question, was a crime committed by the defendant or an unrelated person? It's a binary question, really. If the answer is yes, then the defendant is guilty. If the answer is no, then he or she is innocent. And of course, the forensic scientist would steer clear of this and would very, very rarely consider evidence at this level. But then we come down a level 2, which is called the activity level, and I'll give an example of this later. The question here is, did the defendant or an unrelated person take part in a connected activity, like stabbing an individual, for example? Now, it's possible that scientists may report cases at level 2 if there is sufficient background information provided. Coming down the level this is the source level. Here we ask the question: does the profile from a crime stain, match the suspect or unrelated person? And at the source level, we are generally associating this stain with a body fluid such as blood, semen, or etc. So the scientists would be asking the question, well, does this blood come from this individual? Does this semen come from this individual? And you can do the source level provided there is sufficient material available. But, and as we've seen, when we only have a handful of cells available, we may well not know what the body fluid or the cell type is. And this would be very, very typical in cases where we're examining so-called touch DNA, for example. It's DNA taken from a table surface with no visible body fluid, just cells and sweat, for example. So we won't actually know whether there is blood or semen associated with very, very low levels of DNA. And so we then end up going down to the bottom, which we call sub-level 1. And this is called the subsource level. And this is what we are assessing with the DNA profile. So typically, we've got a low quant value, degraded, small stains, probably partial, no or very little background information, and it's not possible to make an association between the body fluid. Even if blood is detected, it may not be associated with the DNA profile, for example. But this is where the scientist always starts from. We start at sub-level 1 with our DNA profile, and we take it to court. But what the court is doing, the court is always thinking about level 3, and the prosecution will be trying to push the scientist up through the levels to make some comment about the activity level, because this is the sort of information that he needs in order to get a conviction. If the scientist is stuck at sub-level 1, subsource level, and is unable to make any comments about the source or the activity, then effectively the court has quite limited information. It may not have sufficient guidance at the source and the activity level to make a decision about the case. So the point of me doing this is that it's not just the DNA profile that is important here. It's the background information and how we can use that to move up through the hierarchy of propositions. So the hierarchy of propositions framework is extremely important, and we use this as a matter of course in all our interpretation. So, as I've already stated, the strength of the evidence at the DNA profile of subsource level has got nothing to do with the activity level. So it follows then that if I have a statistic of 1 in a billion or 1 in a quadrillion or something like that, then it'd be completely wrong to apply that statistic to an activity. For example, if an individual is accused of handling an object like a knife. And we call this mistake the CSI effect, after numerous TV shows that advertise this kind of thinking. But of course it is completely wrong. So suppose the evidence supports the proposition is 1 in a billion in favour of the prosecution hypothesis. This statistic refers to the strength of the evidence at the subsource level only in the hierarchy of propositions. Now, if we've got a very low level sample of DNA, going back to our 60 picograms, we can't apply the same statistic to the source level. If there is uncertainty that the DNA came from blood, then we can't use the 1 in 1 billion to the source level, because the likelihood ratio must be lower than that. And we certainly cannot apply the same statistic to the activity level. When the activity may be a stabbing, did the suspect assault the victim? So just to show this diagrammatically, the method of transfer is often unknown, especially with low-level samples, and the purpose of the scientist is to explain the limitations of the evidence. So what I like to do is to show a diagram such as this. Here we have the crime event, And this is what we're hoping to achieve. We're hoping that there has been DNA transfer from a perpetrator coincident with the crime event. Of course, this may not be the case. Before the crime event, there may have been an opportunity for adventitious transfer, innocent transfer, especially if an individual has inhabited a premises where the crime was committed. Perhaps they visited it 3 weeks earlier, for example. There's an opportunity for transfer there. And then there's usually a period of time between the crime event and its discovery. After that, investigators arrive. They create the protected scene. They detect and they recover material. It's sent to the laboratory, and it's eventually analysed. And during this time period, there is a potential to contaminate. Again, we're just dealing here with handfuls of cells, and the problem may be that investigators inadvertently move DNA from one location to another, for example, by forgetting to change their latex gloves, which are excellent methods of transferring DNA. And in the laboratory itself, there may be opportunity for the operators to inadvertently contaminate the samples. And the majority of contaminants actually come from laboratory workers and from investigators at the crime scene, and for that reason it's a good idea to have an elimination database of scientists and police investigators on hand, so that the DNA profiles can be cross-compared in order to eliminate these possibilities. And before I move on to the next stage, I want to introduce confirmation bias. Confirmation bias, or confirmatory bias, it's a tendency to search for, interpret, favour and recall information in a way that confirms one's pre-existing beliefs or hypotheses while giving disproportionately less consideration to alternative possibilities. Effectively, what this means is that there is a potential for scientists, investigators, lawyers and judges even to be biased in one direction or another. So the question is, how can we present our evidence in a completely neutral way in order to prevent this bias? Confirmation bias may be completely done at the subconscious level, and this is the danger. Well, we need to be aware of the 3 different routes to transfer a DNA profile. The direct route, that happens at the time of the crime event, for example. The transfer may be indirect or innocent. We call this secondary or even tertiary transfer. And the third method is contamination. And of course, if we have a DNA mixture, it's also very important to realise that the individual contributors to that mixture may be being propagated by different individuals at different times. So we might have one component of the mixture which was indirect secondary transfer, and another component of the mixture which was direct or contamination, etc. But just by looking at the mixture, we don't know which one of these 3 possibilities is true. And there certainly is no date stamp, for example, on the DNA profile. So just to give you an example of a secondary transfer, let's suppose we've got 2 individuals, A and B grabs hold of a knife. The question is whether individual B can transfer DNA from individual A from a handshake. And then let's say this was recovered at a crime scene. We recover DNA from individual A. How confident can we be that individual A actually carried that knife? So let's talk now about an application of the likelihood ratio. Let's just go through an example. So I'll give you the example. This is the case circumstance. A woman is found dead in an apartment. A pathologist confirms death by assault, during which the victim fell She hit her head on a stone fireplace, and she had a massive brain hemorrhage which led to her death. Now, suspicion falls upon an ex-partner who cohabited with the victim up to 4 weeks previous to the offence. Let's say they were in a relationship and he left a month earlier, and he says that he never went back to the apartment. But he is known to have previously threatened the victim. The body is examined, samples are taken, and a mixed DNA profile is recovered from the body of the victim underneath the fingernails, and it matches the ex-partner. So we'll go through the analysis. We start with the subsource propositions. So the prosecution are saying that the DNA came from Mr. Smith and Miss Jones, and the defence say the DNA came from Miss Jones and an individual unrelated to Mr. Smith, an unknown individual unrelated to Mr. Smith. And the scientists can go ahead and give the conclusion at the subsource level with no problem. The evidence is 1 billion times more likely if Mr. Smith and Miss Jones contributed to the sample rather than Miss Jones and an unknown, unrelated individual. So that's all well and good, but to move forward to the activity level proposition, the court must first accept that the subsource evidence is sufficient. And usually if the evidence is overwhelming, billion or whatever, then that is usually sufficient to confirm the subsource. But of course the scientists cannot assume that the court is happy to go along with this, and therefore it's important, before we go to the next stage, to write a caveat that goes into the statement. Because there has to be an acceptance by the court that the DNA came from Mr. Smith before we proceed. So that is what is written into the statement. Provided it is accepted by the court that the DNA came from Mr. Smith, I am able to address the question of the activity. So that it's not the scientist making that decision, it is still the court making that decision. The scientist highlights that before he can go on to talk about activity level, it is imperative that the court accepts that the DNA came from Mr. Smith. If the court does not accept this, then he cannot proceed. So, and just to reiterate, propositions are provided by the court and not the scientist. The scientist does not evaluate the proposition, only the evidence, if the proposition is true. If the circumstances of the case change, or if different propositions are requested by the court, then that means the scientist must carry out a new evaluation of the evidence, and then that would usually be made clear in the statement as well. The scientist has to be extremely clear about what he is basing his evaluation on, what the assumptions are. So these are the activity level propositions now, and again this is the information provided to the scientist by the court or the court representatives. So the prosecution say Miss Jones scratched Mr. Smith on November the 21st, which was when she was murdered. The alternate proposition by the defence is that Miss Jones did not scratch Mr. Smith on November the 21st and has no social interaction with her since October the 21st. He did not assault her. So these propositions outline the differences of opinion between the prosecution and the defence. And it's also important to note that the propositions here, they do not mention transfer of DNA. And this is because we must not mix the propositions and the results together. The propositions are formulated to describe the points of differences between the prosecution and the defence. And you can see, hopefully, that these propositions can be formed independent of any results. These propositions don't depend on any knowledge of results, and this assists in preventing confirmation bias. Often the propositions in casework may be unclear, and there is no reason why multiple sets of propositions may be formulated. So we may be dealing with several sets of propositions. So what are the considerations? Well, if the first proposition is true, then the DNA recovered is a result of direct transfer. It must have happened at the time of the offence. And now the scientists must start to think about transfer, persistence and recovery of DNA. It's not just transfer of DNA. We've got to think about, well, has it transferred? What's the likelihood that it will persist on a particular surface? And what's the likelihood that I will recover it? So we often talk about DNA transfer, but what we really mean by that is what is the probability of transfer, persistence and recovery of DNA if Miss Jones scratched Mr. Smith. And of course the alternative is what is the probability of transfer, persistence and recovery of DNA if Miss Jones had not scratched Mr. Smith. Now let's just look at the defence proposition. Now clearly, if Mr. Smith is innocent, then the question that remains is, well, will the DNA persist for 4 weeks in the apartment? Can it be transferred to the victim by secondary transfer? This is innocent transfer. And will it be recovered upon analysis? So really those are the three questions which the scientist has to deal with. And typically at this point, the scientist might be asked by the court to express an opinion based upon his or her experience, and the scientist may opine something like, I've had 30 years experience and I've never seen an example of secondary transfer 4 weeks after the last known contact. Now, there is a problem with this kind of assertion, and the problem is that it is based on intuition, and intuition is certainly a very poor substitute for doing experiments, and intuition can often be wrong. And by that I mean that it's subjective. These kinds of statements can be prone to confirmation bias. They're not objective. They're based on casework experience, and the problem with casework experience is that we can never know the ground truth of the cases that we have reported. And because of that, there may be serious risks of misleading the court, resulting in potential miscarriages of justice. And this kind of evidence, it can't really be properly tested by the defence either. There's nothing really for them to get their teeth into. So, to summarise, courts should not rely on experience. Instead, scientists must rely upon hard data to support conclusions so that we have transparency. And to do this, the court needs to know what experiments have been carried out in order to form a view of the strength of the evidence. Ideally, these experiments will be published and peer-reviewed in the scientific literature. And if not, then there should be experimental data available. Maybe in the lab has carried out some experiments, and this should be available for disclosure so that it can be scrutinised and challenged if necessary. And of course there may be experiments which have been carried out, but the court will want to know if the data are relevant to the case in hand. One of the problems of this, of course, is that every case is unique. The circumstances are unique, so the question will be, well, whether the experiments which have been carried out, are they actually relevant to the case in hand? In any event, the scientist should always represent the consensus view of a community of scientists. I would argue that personal views should not be given unless they are based upon hard data. And I would discourage personal views which are based upon subjective opinions of a single laboratory. The problem with relying on views of a single laboratory is that we may generate a pseudo consensus where Individuals in one laboratory may report one way, individuals in another laboratory may report another way. And I call this pseudo-consensus. And we saw an example that Tamra spoke about with Mix 13, which illustrates this problem with mixture interpretation, where laboratories use different protocols. So what kinds of experiments are needed? So if I just continue with the example I gave, we could set pairs of individuals to scratch each other. Obviously, we're not allowed to violently scratch individuals, so inevitably there will be light scratching of individuals. The samples will be taken from different time individuals from the person who's been doing the scratching. And if we do that, then we can answer questions like, well, how long does DNA persist for? We can take samples of 1 hour, 2 hours, 3 hours, 4 days, etc. But of course, we have to consider many other factors like washing. Did they wash themselves? Their shedder status. Some people shed more DNA than others, etc. And I think it's also important to take controls from crime scenes, so we get an idea of the background DNA present at the crime scene. In the example I gave you, does the individuals, the suspect's DNA exist in areas which are not associated with the offence, for example? And these experiments go forward to tell us or give us information about how long DNA persists in the environment. So we need to carry out experiments to evaluate the presence of background DNA. Background DNA is DNA from unknown individuals, and we need to find how long prevalent DNA exists. Prevalent DNA is DNA from known individuals. We need to carry out controlled experiments to determine how long DNA persists when someone leaves the premises. In my example, the individual had left the premises and never returned in a period of 1 month, according to the defence proposition. And there are other factors to consider, for example, whether the premises have been cleaned in the interim period, etc. Did the owner of the premises clean every day? Did she use bleach? Or all these factors need to be considered. We need to think about how easy is it for DNA to secondary or tertiary transfer, and we can carry out quite simple experiments to detect transfer, secondary transfer in the laboratory. So, for example, individual A holds a tube. Some hours later, individual B holds the same tube, and we take a swab from individual B to see if DNA of individual A persists. And we can repeat these experiments numerous times, varying the time intervals of sampling. So I don't have time to talk any more about these experiments, but experiments such as I've described have been carried out, and this would give us sufficient information to produce a likelihood ratio at the activity level. And a typical statement at the activity level would then follow: Miss Jones scratched Mr. Smith on November the 21st. The alternative proposition: Miss Jones did not scratch Mr. Smith. He cohabited until October the 21st. I've already told you that a DNA profile was recovered and the probability of the evidence at the subsource level was a billion. And if I calculate a likelihood ratio under typical circumstances with this scenario, however, I may well come up with a very, very low likelihood ratio, and the statement would follow under the assumptions listed above. From experimental evidence, the evidence is between 12 to 40 times more likely if the first proposition is true. Now, this isn't based on a real case, but this level of likelihood ratio, the activity level, is not atypical. Often these levels are extremely low. And of course, the point that I want to make is that these activity levels, propositions, and the likelihood ratios results in extremely low likelihood ratios compared to the likelihood ratios at the subsource level. And I make this point to illustrate the dangers of carrying over the 1 in a billion likelihood ratio at the subsource level to the activity level. So I think it is really important that the scientist always takes some time to explain to the court that his 1 in a billion statistic only refers to the DNA profile. It has absolutely nothing to do with the activity level likelihood ratio. So the importance of the hierarchy of propositions is that each level of the hierarchy of propositions has its own likelihood ratio. And of course, whether the scientist can calculate these likelihood ratios is a different question, of course. But in relation to that, we must note that DNA is very stable and it does persist in the environment for a long time, especially if that environment is dry. And to conclude, I just want you to note the contrast, the strength of the evidence in the activity statement compared to the subsource statement. Now, you may not have heard very much information about what I've just been speaking, and that's because there isn't so much research which is carried out in this area, and that's why there is limited information on these kinds of issues. And certainly there is a need for more funding and better communication between the scientist, the court and the investigators. But if we do not think about these issues, there are a number of dangers that may arise. Now let's suppose the scientist goes to court, the scientist just reports at the subsource level, and effectively if the evidence finishes there, then the court is left to its own devices to make a decision about the activity level. And of course, this is the main problem, I think, is that the ill-informed court may carry over the subsource likelihood ratio to the activity level. And this would inevitably lead to miscarriages of justice. So I think it is not an option for the activity level to be ignored. Just because it's difficult doesn't mean to say that we can't discuss it. The scientist must alert the court to the dangers of relying upon the subsource statistic. So what the scientist does is adds value to the interpretation of the evidence by following the framework of the propositions and by considering the likelihood ratios in the context of the activity level. The scientist must be aware of the background information in a case. Communication, of course, is paramount. So to summarise, appreciation of the hierarchy of propositions places the evidence into context. We should avoid what I call the association fallacy, where a DNA profile may be inappropriately associated with something else, like a body fluid or activity. And I think we need to recognise amongst ourselves that confirmation bias can have a significant effect on the objective interpretation of evidence. And to illustrate this, I now want to turn to a real case which I was involved with, and this is the case of miscarriage of justice of Amanda Knox and Raffaele Sollecito. I'm not going to go through the judgments, but it was an extremely long affair beginning in 2009 and 2015, where the defendants were found guilty, and then they were exonerated, and then they were found guilty again, and then eventually the exoneration was upheld. We've got to remember that the Italian court is inquisitorial as well, and the court process is very different to that which we experience in adversary courts. I'm sure we're all aware that there was a subject of immense media scrutiny. I think once miscarriages of justice are identified, however, it's really important to analyse them so we can identify flawed practice that led to the wrongful convictions. We need to consider what could be done to prevent reoccurrence in the future. we need to think about how the interaction of scientists, investigators and judges can be improved. And equally importantly, when miscarriages of justice occur, they often reveal a way of thinking. And because of this, this is not the only case in which this thinking has been used. So it leads to the question, that are there other cases which are already prosecuted that are similarly affected, where these individuals may be, of course, wrongly convicted. So I'll go through the case circumstances. In November 2007, Meredith Kircher was brutally murdered in her bedroom. She was stabbed in the neck. She shared the flat with Amanda Knox and other students. The prime suspect was Rudy Guerdi. He was quickly captured. His DNA was discovered at numerous locations in the crime scene. And what I want to highlight here, that crucially, he did not have legitimate access to the crime scene. But he implicated Knox and Sollecito as collaborators, and this is the reason they became suspects. But of course, Knox lived in the apartment. Sollecito was her boyfriend, and he had already had been to the apartment prior to the murder. But the prosecution continued because the prosecutors insisted that the prime suspect, Gueredi, could not have acted alone. Now, the DNA evidence, three crucial parts to this. I won't have time to go through all of them, but I'm going to talk about the knife which was recovered from Sollecito's flat, which was used to implicate Knox. There was a bra clasp, which I haven't got time to talk about, and blood spots in the bathroom, which I will talk briefly about. Okay, so here is a rather large knife, which was actually recovered from the Sollecito's cutlery drawer from his apartment. The DNA profiles were obtained from here, A, B. This area revealed a DNA profile which matched Amanda Knox, and there was a very faint profile on the blade which matched the victim. The profile attributed to Amanda Knox, it was a good quality profile, which is illustrated, and the origin of this DNA was not disputed. But on the knife blade, it was a very poor quality profile. When I analysed this, I got a likelihood ratio in the region of about 100, so the evidence is quite weak. Now, the collection of the knife is as follows. It was found in Sollecito's flat, and it was in a cutlery drawer. The knife was observed to be clean. The investigators noted that there was a strong smell of bleach, so they hypothesised that the knife had been cleaned with this bleach to remove blood, because obviously if a knife has been used in a murder, you would expect it to be covered with blood, but there was no sign of blood whatsoever on this knife. Amanda Knox's DNA profile was detected on the handle of the knife. A very weak profile was attributed to the victim. All the tests for blood were negative. So my comment here is the interpretation is currently at the subsource level. We have two DNA profiles. This interpretation is at the subsource level. There's no direct evidence to determine the body fluid origin, noting that if the DNA came from the victim, it must have come from blood, according to the prosecution hypothesis, of course. But there is no evidence here to suggest that this DNA did come from blood. There is no evidence to suggest the activity of transfer, therefore. So we are firmly in the subsource level. Now, the investigators' hypothesis of bleach cleaning, this is an example of confirmation bias. It's very inconvenient to the prosecution, of course, not to have blood on a knife, and therefore they're thinking of ways to explain this. which is why they come up with the bleach cleaning hypothesis. But this falls very firmly into my definition of confirmation bias, where prosecutors are using information in a biased way to explain lack of evidence. This case actually had a pretty good defence on it, and the defence examined the knife. They observed starch grains. So if we are now working through the hierarchy of propositions framework, we can start to come up with alternative propositions. I hasten to add that at the actual trial, this process was not followed, and what I'm doing is showing how the process should be followed if I was doing this particular case. So we can ask two alternative propositions. Either the knife was used in the murder, or the knife was only used by Knox to prepare food. Those are the two alternatives that the prosecution and the defence are putting forward, because it's known that Knox visited her boyfriend quite often, and she used to cut bread to make food. And so this seems to be a possible transfer method. Now, interestingly, the Massai motivation, this is what one of the judges said in the original prosecution. He stated, "It appears more likely to have been derived from her having held the knife to strike rather than from having used it to cut some food." So for some reason, the judge, after hearing the evidence, seemed to be able, from the mere presence of the DNA profile, to say that she used the knife to strike the victim rather than using it to cut some food. But I can tell you now that this was pure speculation, and there is no way that we can actually do experiments to show the difference on the knife handle, to show whether a perpetrator is holding it to strike an individual as opposed to cutting food, because our expectations of the DNA profile, the distributions of the DNA profile, are identical. So there is really no basis for this. But yet the statement was made and the individuals were found guilty. And now I want to contrast that with Rudy Guerreri, the prime suspect. He had no legitimate reason to be in the apartment. His DNA was found in multiple locations in the murder room and in the bathroom. Even though Knox lived in the apartment, her DNA was not found in the murder room. So there's complete contrast in the distribution of DNA profiles here, and this implicates this distribution of DNA certainly does implicate Guerreri. The crucial point being that he had no legitimate reason to be in the apartment, and therefore it is actually difficult to think of an innocent reason for his DNA being present in so many locations. The only DNA attributed to Sledgeto was a minor profile found on a bra clasp. Unfortunately, I don't have time to talk about the bra clasp, but I do want to talk about mixtures of DNA found in the bathroom. Mixtures of DNA from Knox and Kircher, the suspects and the victim, were found in the bathroom, and it was assumed by prosecutors to indicate that Knox had washed her hands after the murder. And so again, we see an example of confirmation bias. They postulated that the presence of DNA in blood stains, mixtures of Knox and Kircher, must have showed that she had washed her hands after the murder to get rid of the blood. However, because these individuals had been cohabiting, we do have a prior expectation that DNA from cohabitees will be widely found together throughout shared premises. And this DNA comes from skin cells, sweat, saliva, from talking even. So especially if you share a bathroom, I can guarantee that your DNA will be everywhere in that bathroom, along with other people who share it. And here is an example here. This is, you can see the blood stains here. So the blood stain was taken and analysed, and Mixed profiles were obtained of victim and Knox, and therefore the prosecutors assumed that this was a mixture of blood from the victim and Knox. But what we have here are two separate tests. We have a test for blood, we have a test for DNA. Now, just because we test positive for blood doesn't mean to say that the DNA, which have come from the same location, has all come from blood. What it does probably mean in this case is that the blood has come from the victim, but the DNA attributed to Amanda Knox has just come from skin cells. We cannot attribute the DNA from Amanda Knox to blood. Even if there is a positive result for blood in that stain, simply because it could have been deposited earlier. So, more examples of confirmation bias. There were many stains where the DNA matched the prime suspects. The lack of DNA in the victim's bedroom was bizarrely explained away by prosecutors by selective cleaning hypothesis. They actually proposed that Knox and Solecito clean the flat to remove their own DNA, leaving behind that of Guedes in order to implicate him. But of course, this is something which is completely impossible. You can't selectively remove DNA from a room in order to leave a DNA from another individual. Fortunately, we got to a position after several years in the final judgment where the individuals were exonerated, where these arguments were put forward to the judge. And the judge agreed that the confiscated knife could have been moved by Knox to Via della Pergola for domestic use on the occasion of parties or other events, and thus also used by Kircher. A lot of problems with this case. The collection was seriously criticised. When the knife was collected, it was put into a common cardboard box of the sort used to package Christmas gadgets. It was also demonstrated when the crime scene was investigated that investigators failed to change their latex gloves, therefore providing an opportunity of transferring DNA from one location to another, because it's been shown that latex gloves are excellent means of transferring DNA. And finally, the bleach cleaning hypothesis was dismissed as completely illogical. What this goes to show, once the crime scene is compromised, it's irreversible. Nothing more can be done, as happened in this particular case. And the final judgment said the use of logic and intuition cannot in any way compensate for the lack of evidence or the inefficiency of investigations. Faced with missing, insufficient or contradictory evidence, the judge should simply accept it and issue a verdict of acquittal. And there is a plea for objectivity in scientific investigation, following the scientific method, starting with Galileo Galilei on the application of the scientific method. So in this case, it is possible to define the reasons for miscarriages of justice. Certainly the prosecutors were working backwards, they were fitting the evidence to the crime. Inconvenient results were explained away by bizarre hypotheses, and the bleach cleaning hypothesis and the selective cleaning hypothesis. There was certainly a failure of judges to take adequate account of defence criticisms. As I said, there was quite a good defence in this case, but the judges didn't take full account of this. Remember, this is an inquisitorial system. There are no juries involved. And there was also a failure of basic quality standards in collecting the evidence, which resulted in contamination of the crime scene, by which I mean the contamination or the probable contamination using the latex gloves. And before I move on to the next part of my talk, I just want to draw your attention to this document. This was prepared under the funding body which I'm a member of, the Euro4Gen Network of Excellence. It's funded by the European Union, and we produced this booklet which you can download, and that goes through a lot of the material which I've been discussing. So to conclude, prosecutions using DNA alone are likely to be unsafe, and that's because it seems inevitable the strength of the DNA evidence is transposed to the activity level. So what I've shown is that really we need to operate within a framework, but for this framework to work properly, the court must provide a suitable background for this to occur. I think it's important defence scientists must be educated to the same level as prosecution scientists, and they need to have access to equivalent facilities to carry out tests. In particular, lawyers and judges must be sufficiently educated so that they understand the hierarchy of propositions framework and are able to apply it properly. And they need to avoid pitfalls like the association fallacy or the prosecutor's fallacy. So that concludes my part of this part of the talk. But what I want to do now is to move on to interpretation at the subsource level. I'm going back now to the DNA level. And I want to complement Tamara's talk with a brief introduction to low template DNA. There will be some repetition here from Tamara's talk, so I can go through that fairly quickly, I think. Let's go right back to the beginning. Well, DNA profiling in forensic science was first demonstrated more than 30 years ago, and these original techniques required a lot of material. You needed at least a square centimetre of blood, and they weren't very sensitive. So if we fast forward 30 years, we see now that we have very increased levels of sensitivity thanks to low copy number profiling using low LC, using low template profiling. And in fact, we're able to detect just a single cell if we want to. Now, this sensitivity method extends the utility of DNA profiling. We can get DNA profiles from materials which we couldn't dream of 30 years ago. We can get DNA profiles from skin cells. And what this illustrates, as I've explained, is that with this increased sensitivity, there is a downside, and to manage the interpretation, we need a framework to interpret the evidence. So my talk now just discusses interpretation of the DNA profile at the subsource level only. So, as Tamara has already pointed out, this is what we hope to see when we carry out a DNA profiling. We want to see a good DNA profile. It's well balanced, the artifacts are low level, and there is sufficient quantity of DNA to visualise. In this particular example, it's not a mixture because we just have 2 alleles per locus in this example. But we can see here, this is the reference sample, and this is the crime stain. We can see that the reference profile does not match the crime stain because we have imbalance. The peak heights of the crime stain are much lower, which results in poor balance. And not only that, we have a phenomenon where alleles may be missing, and this is called dropout. So we've got an example here, missing allele here, missing allele here, an example of a low level allele which may be unreported because it's too low. And again, Tamara has already pointed out the effect of degradation. If we do an experiment, we take a reference profile and we put it in a damp, humid box for a number of days. It gradually disappears. So the high molecular weight alleles disappear before the low molecular weight alleles. Now, this is a classic cause. Degradation is extremely common in casework. And the first characterization of a low template DNA was carried out by myself and colleagues in the Forensic Science Service in the year year 2000. And in this paper, we highlighted two main effects, drop out and drop in. And we discovered drop in by simply looking at controls, negative controls. Now, obviously, negative controls should be completely free of DNA. But when you carry out low template DNA, they're not. And this is the first example that we came across, that we published in the paper. We've got 30 samples here, and if we analyse these 30 samples, we see random alleles appearing at random loci, usually just one or two, no more than two or three in this early technique. Actually, more modern techniques, we wouldn't expect to see this level of drop-in, but this was early days. So it follows that if I have drop-in going on in my negative controls, what this means is that it must be happening in my case samples as well. But what is drop-in? Well, drop-in is probably just fragments of DNA floating around in the atmosphere, falling into the tube. Household dust, for example, contains fragments of DNA chromosomes, and occasionally these fragments are going to fall into tubes and they're going to be analysed. Remembering that I said that the technique is very, very sensitive down to just a handful of cells. So this is going to happen, and we can't avoid it. But what we can do is to take it into account when I calculate a strength of evidence. So this phenomenon of drop-in is not a disadvantage, because we can take account of it when we carry out probabilistic genotyping. What we do need to do, however, in our laboratory is to carry out experiments to work out the levels of drop-in which we expect to see in our negative controls. Now let's just look at mixtures. Often we don't have simple profiles from a single individual. So this is a single locus from an individual. This is not too much of an interpretation issue. We've got 4 alleles, and let's say we've got a major contributor, type 16, 18, and a minor contributor, type 14, 17. And if we analyse a reference sample, we might find that the major contributor is 16, 18, which means that there is an unknown individual under the prosecution hypothesis here and here. But when we have this kind of separation, the contributors are easily separated. We've got good levels here, good peak heights. So it's very unlikely that these alleles will be associated with each other. And we can confidently separate these out. In addition to that, we can work out a mixture ratio very easily. Mixture ratios are important to probabilistic genotyping. Often it's not as easy to work out as it is here. And if we go through the calculation just by adding these two peak heights and adding these two peak heights and dividing one by the other, then our ratio in this particular example is about 1 to 3. So here we have the same mixture, looking at different loci, 2 individuals. It's more difficult to interpret when there are 3 or 2 alleles present, because the contributors share alleles. So in this example, we just got 2 alleles. We're postulating that there are two contributors here, and if that is true, then the contributors must be sharing alleles. For example, if this individual, the crime stain is 811, it means that we've got a homozygote. We could have one individual who's a homozygote 1111 and another individual who's a heterozygote 811. These peak heights are always additive, and if this is the case, then this explains why we have this imbalance here. It's because we have this homozygote 1111 added to the heterozygote 811 on the allele here. So to interpret, we use the likelihood ratio method, as I've explained. The prosecution say the DNA's come from Mr. X and an unknown person, and the defence say the DNA has come from two unknown persons. And as I pointed out, with the major minor profile with 4 alleles, the calculation is straightforward. Not so straightforward with the 2-person mixture, it's more complicated. So if we ascertain that the suspect is type 811, the prosecution proposition is the DNA is from the suspect and an unknown individual. Now, under the prosecution hypothesis, we have to go through all of the alternatives. The unknown individual must be type 811, 88 or 1111. Those are all the possible alternatives. The defence proposition is more complex. The defence say it's from 2 unknown individuals, hence the alternatives must be more extensive. So the defence will say, well, the minor contributors could be 88, the major could be 1111, 88, 811, 1111, 811. Or they could be 811, 811. And in addition, we need to swap these round, so there will be a few more possibilities to consider. Mixture ratios, as I showed you earlier, help to assign a weighting or likelihood of each combination, and that can be used to calculate the probability of the evidence. Now, the more contributors we have in a mixture, the more complicated it becomes, of course, because we might see 6 or more alleles at each locus in a DNA profile, and we need to work out all of the possible pairwise combinations. So, whereas I can carry out a preliminary analysis using an Excel spreadsheet, for example, as soon as we start getting into these complex scenarios, we do need a computer to carry out the calculations that are very, very extensive. Fortunately, the calculations are very repetitive and they follow a series of well-established rules, and for this reason, it's relatively straightforward to produce probabilistic genotyping software. And of course, with low template, we can't really rely on heterozygote balance. It becomes much less predictable. It becomes difficult to separate the major minor contributors that I showed you earlier on. And as Tamara pointed out, with major minor mixtures, the minor contributor alleles may not be distinguished from stutters. And so the problem is, as Tamara's already explained, that if we have a major allele, parent allele, a portion of that allele can be moved over here, maybe it will be 10 or 12% or whatever the validation has shown, and then we get a minor allele here. Stutters fall in allelic positions. We can't tell the difference between a stutter and an allele, so the temptation is to filter them out and ignore them. But of course, if the person of interest is a minor contributor, then it may be the same level as the stutter, and indeed this may be a mixture of a stutter and an allele. So we're getting into quite complex scenarios here, and the only way to resolve these issues is using probabilistic genotyping. And really just to illustrate how we get to dropout. Dropout is related to heterozygote balance. So here we have a perfectly balanced DNA profile. If we reduce the proportion of the DNA, reduce the level, go down to 30 picograms, eventually it becomes unbalanced and eventually we have dropout. So I've talked about dropout and drop-in. So let's suppose that we have a crime stain, we're just looking at one locus, and let's also, just for context, assume that the rest of the crime stain looks okay, it matches. But suppose we have a single locus that looks like this. The crime stain has got a peak here at A and one at C here. And then we look at the DNA profile from the reference sample from the suspect. We find it's an AB. Now, this is not an exclusion. How come? Well, we need to go through a logical process. The prosecution, if this is true, under the prosecution hypothesis, then allele A is present, there is no dropout. But there is dropout of allele B, because allele B is missing. And therefore, if the prosecution hypothesis is true, allele B has dropped out. And then we've got this allele C over here. If the prosecution hypothesis is true, then C is a drop-in. Now, of course, the defence alternative is quite straightforward. They just say there is no dropout, there is no drop-in, the unknown is type AC. The unknown is type AC. So it's fairly straightforward. Now, it's not an exclusion, and I wouldn't describe it as neutral, but the likelihood ratio will be less than 1. But the point is, as I come back to the first talk, was that the likelihood ratio is unbiased. It is possible to calculate likelihood ratios that favour the defence hypothesis or the defence proposition of exclusion. Now, if the rest of the profile is okay, it's matching, then that will counterbalance this, and the likelihood ratio will be greater than 1. But the point is that when we use probabilistic genotyping, we no longer need to succumb to the temptation to ignore inconvenient loci. And of course, it's wrong to ignore inconvenient loci because the likelihood ratio will be less than 1 for this example, and it would inevitably reduce the overall likelihood ratio. So it is never wise, I think, to ignore evidence for that reason. You're not necessarily being neutral. You can be anti-conservative and biased. So it's important to use the likelihood ratio. It's the only way to properly assess the evidence. Its use is dependent upon propositions formulated by the prosecution and the defence. The propositions are based on background information of the case. It's okay to change the propositions in the investigative phase. We can always update with new information, but ideally the proposition should become fixed in the evaluative phase when we go to court. And that's simply because the process becomes unmanageable. If you're giving evidence in court and the defence come up with different propositions, then effectively you have to go away and do a different analysis. So that's one reason why we have to put caveats in our statements to alert the court to the point that if it is necessary for me to evaluate different propositions, then this will take some time to do. There's nothing worse than trying to do this on the hoof. The likelihood ratios for subsource level need to include the number of contributors, and also we need to know the known individuals as well for conditioning under the defence and prosecution propositions. Court engagement now. I want to talk about the court process for giving evidence. And there are, as we've seen, a number of different computer models that are in existence. And the same is exactly true in Europe. There's quite a few different models, maybe up to half a dozen different models are routinely used in casework. But the more advanced models certainly take account of the principles which I've described so far. They accommodate multiple contributor. In my opinion, there's no realistic possibility of standardising onto a particular model, and therefore it becomes important to compare results between different models. But models, I think we can conclude that models that operate on similar principles should provide similar results. But there are inevitably some court responsibilities to consider. The interpretation of evidence is complex, as we've shown. We've got to be aware of the hierarchy of propositions. We've got to be aware of limitations. And I've talked about the dangers of carryover. as part of this, the defence have an important role in setting the propositions. And I know that often scientists working for the prosecution have limited or no interaction with the defence, but for this framework to work properly, then we really do need to think about the propositions that the defence want to put forward. So I would argue that scientists operating for the defence do certainly need to be educated to equivalent levels of the prosecution scientists so that this communication can occur. They need training and they need access to the tools. The court role, I believe, is to facilitate the discussion about the case using the likelihood ratio framework in an unbiased way. And certainly judges and lawyers also need to be educated to ensure that they are fully cognizant, and they themselves need to avoid confirmation bias, etc. So, and added to that, there is no preferred or standard model or true model. There is no such thing as a true model. There is no such thing as a true likelihood ratio. Not all models that we use are freely available, and there may be restrictions on its use, and therefore it's inevitable that prosecution and defence scientists will use different models to evaluate the strength of the evidence. The likelihoods depend upon the modelling assumptions, as I've indicated earlier, and as I've already said, the most important aspect, I think, there is no true model, and therefore it follows there is no true likelihood ratio. And I know from experience that this does actually cause some confusion in court. It's not like measuring a piece of string, is one analogy which has been used. So it follows that different models will provide different LRs because they will use different modelling assumptions. And even if the data in the prosecution and defence propositions are identical to both, even if the models are validated to court-going experiences, they will inevitably produce different likelihood ratios. But what we can conclude, provided the models are valid and shown to be valid, that these different answers are both expected, and even if they are different answers, they are valid. Both answers are valid, even if they are different. So there is an inevitable outcome in the adversary court, is that reports will be routinely prepared and challenged by different software that use different modelling assumptions. Of course, there is a great benefit to this. If similar answers are obtained, then confidence in results should increase. But if there is a great difference in the answers, then the matter needs to be properly investigated to discover the reasons for the discrepancies. And following a paper from Steele and Bording, who have carried out some comparative studies, they state that we would expect to see an order of magnitude difference in the answer. It's not unusual. So I want to talk about a case that I was involved with in the UK, where the prosecution and the defence used alternative methods. And method A gave a result of 2.9 million. It was a partial profile prosecution. But when the defence carried out the analysis using a different software, they came up with 300,000. The defence also carried out a different calculation using different assumptions, and they came up with a figure of 40,000. So they came up with two different figures. So of course the 40,000 is very different from 2.9 million. 300,000 is less than 2.9 million, but it's within the order of magnitude limit. Now, it's inevitable that courts will find this quite difficult to deal with, because what we're telling them is both these numbers are valid given the assumptions, given the models have been validated for court-going purposes. So the judge in this case ordered that the experts for the prosecution and defence met in order to prepare a joint statement. And we have a rule in the UK, it's Rule 33.6, which is specifically for this purpose. The court doesn't want to get involved with disputes if the scientists can resolve them. So what Rule 33.6 says, it will be necessary that the court directs a meeting of experts so that a statement can be prepared of the areas of agreement and disagreement. Of course, so the court is only interested in the areas of disagreement. No lawyers are present during these meetings. But of course, it does mean that the defence and the prosecution must be willing to collaborate with each other. Of course, it goes without saying. So in this particular case, the prosecution and the defence collaborated, and they agreed the propositions to test. Because, as I said, in the original trial, the defence had offered two alternative sets of propositions to test. But after discussion, the defence agreed with the prosecution to test these propositions. One, Mr. X, who is the suspect, and two unknown and unrelated individuals, and the defence proposition is three unknown individuals unrelated to Mr. X. So these sets of propositions here just highlight the differences of opinion. Well, just one difference of opinion. Mr. X is either present or he is absent. And then the analysis was carried out. It was exactly the same as before. Method 1 gave 2.9 million. Method 2 gave 300,000. So in the statement, we wrote, Steele and Balding suggested a difference of about one order of magnitude, e.g. 300,000 versus 3 million, is negligible. To aid understanding and to assist the court, we suggest that it may be preferable to convert the numeric strength of evidence into a verbal scale, which operates on an order of magnitude basis. This has been proposed by the European Network of Forensic Science Institutes and is reproduced in its entirety below. So if we use the verbal scale, we'll give the court the scale in its entirety. And then the statement conclusion simply goes, therefore, in our opinion, the forensic findings provide very strong to extremely strong support for the proposition that the DNA profiles originated from Mr. X and two unknown unrelated individuals rather than from three unknown contributors who are unrelated to Mr. X. This is possibly preferable to expressing the evidence as numbers, but of course we always give numbers along with the verbal scale. Not the other way around. If we give a verbal scale, we would always furnish it with a number. And a final conclusion. I think this is my last slide. It is important that the technology is not just viewed as a prosecution tool. It is important, I think, that defence and prosecution must have access and must be able to engage in a meaningful dialogue. Otherwise, inevitably, with this technology, courts will become confused. And as I've outlined in red, we do need to move to a more collaborative style in court rather than a combative attitude between experts. Otherwise, I think it will not work. Crucially, don't expect the same answers from different models. Courts will need to adjust their expectations. And most importantly, do not confuse the subsource LR with the activity LR. And that is the end of my talk. Thank you very much.
Thank you, Peter. Thank you so much for your talk. We have under 15 minutes left before Lynn's talk, so maybe we'll go through a couple questions if that's okay. We have, is that okay, Peter? We've got
Yes.
a list of questions for you, and I think what I'll do is share my screen. Let's see if I can. Let's see. I'll share my screen because I can then you can then read the questions as well. Okay.
Okay.
The first question. Okay. The first question: do you believe that the use of terms such as touch DNA contribute to misconceptions of propositions as pertains to the hierarchy? Our agency has tried to educate the prosecutors and have requested that they refrain from referring to trace DNA as touch in the courtroom.
Yes, I completely agree with that. I shouldn't have used the word touch. I should use the word trace. So yes, I agree with that.
Okay. Next, thanks for mentioning the transpose conditional. Can you comment on the defense fallacy?
Yes, the defense fallacy is where the defence come along and say that there's say 1 in 500 chance that the individual is innocent rather than guilty. Also,
Okay, yeah.
yeah, but it's really the defence fallacy really just takes the probability and gives a probability of innocence.
We actually have Stephen Myers coming up in one of the lectures. He's going to be giving a talk near the end of the series specifically on the fallacies and listening out for those differences, so that will help as well. The hierarchy of propositions guidelines being practical. When was this guidance created or adopted for European labs?
Well, this has been around for quite a few years. I think the hierarchy was actually proposed about 20 years ago, and unfortunately in our field it does take a very long time for ideas to actually get used in practice. I think I have to say that the reporting at the activity level is not practiced very much in laboratories. A few laboratories in Europe will practice this, but there is a problem, I think, with funding to carry out the experiments and also education. We are publishing guidelines under the auspices of the ISFG. There is one set of guidelines which is out, and there will be guidelines on activity level which will be published in the coming months.
Okay, great. How would the transfer events be built into a proposition? I think you gave one example that has a different.
Yeah, I did give an example. We try to avoid using the word transfer in the proposition. The example that I gave was based on DNA under fingernails. So either the individual scratched someone, or there was or the DNA came from secondary transfer. So the propositions would be individual A scratched individual B, or individual A did not scratch individual B. We don't talk about transfer as such. We are talking about the events which are happening. But we do need to consider the transfer when we calculate the probabilities. However, it's not straightforward, which is why there does need to be educational programs to explain.
Okay. How can we be sure that control samples from crime scenes are in fact controls?
Maybe it was a wrong choice of words on my part, but the idea of taking samples from a crime scene, we need to try to establish the levels of background DNA if we can, and we need to try to establish the levels of DNA from known individuals. I suppose in the strictest sense they are not controls, but we do need to somehow get an impression of the overall background of DNA at a crime scene. With the example that I gave you, the individual who was accused said that he had not been to the premises for a period of 1 month. Yet his DNA has been found on underneath the fingernails. Now, I would argue that it would actually be quite useful to sample the premises to find out the levels of his DNA which are prevalent. If there is no, for example, if you do some sampling at the crime scene and you find there was no DNA from him at all, then I would argue that actually increases the strength of the evidence in favour of the prosecution hypothesis. But if his DNA is everywhere, then it is then that would tend to favour the defence hypothesis.
This makes me think about the sink swabbing that you showed. I don't know where the sample was collected, but if it was collected from, say, that stained area, I mean, you might think from using the sink that that there was background DNA if another area of the sink bowl had been swabbed outside of the blood stain.
The difficulty with the Amanda Knox case is there is no time difference. Amanda Knox didn't leave the apartment a month earlier. They were living together at the time, and that means that you have a prior expectation of her DNA being everywhere in the apartment. In that respect, you don't really need to do experiments to come to that kind of conclusion.
True. Okay. Let's see. Your next question was, is the PG software available, capable of answering questions related to transfer or activity without the experiments that you've called for?
Well, there is software which is published, and it's usually freely available. It's not like carrying out probabilistic genotyping. So there is no probabilistic genotyping for a transfer activity. It's a different program. These programs are called Bayes nets, and fortunately they are much simpler to construct compared with probabilistic genotyping software. And usually there is no issue with accessing this software. However, the software can only work if you inform the probabilities with experiments. So there is no getting away from doing experiments. There are certainly not enough experiments which have been carried out, and what we really need are knowledge bases. That is where these would be repositories of individuals who have carried out experiments and deposited them on an accessible database. If we had this, then we could quite easily use quite simple software solutions actually to carry out an analysis of these questions of transfer or activity.
Okay. You have one last question, and it's a good question. We've considered, you spoke of the defense proposition being unknown, unrelated individual. What if the defense is that it's not me, it's my brother? How does that change the likelihood?
I think the majority of probabilistic genotyping software are able to take account of this. What it will do, it will considerably reduce the likelihood ratio calculation. The propositions are entirely different, of course. The prosecution are saying that it's a suspect, and the defence are saying that it's his brother. The prosecution aren't saying it's anyone else. It's important to note that the prosecution are not saying it's a random individual. So what you effectively do when you go down this route is your population of suspects is effectively reduced to two. It's either the suspect or the brother. There's no other consideration in this hypothesis. The likelihood ratio will be considerably reduced. Of course, the best thing to do is always to find the brother and analyse him separately, and that would be conclusive.
Yes. Okay. Well.
Looks like we might have dropped Tamara momentarily. But in the meantime, Lynn, if you would like to go ahead and start your presentation, you can go ahead and grab control of the screen.
Okay, I'm doing that now. Okay. Thank you, everyone, for sticking with us this long. The talks from Dr. Moretti and Dr. Gill were fascinating for me as an attorney. We'll be shifting focus a little bit for this last session because I am a lawyer and I'm an attorney who doesn't actually try cases day in and day out. But because I'm with the Texas Forensic Science Commission, I see a lot of these issues up close. And Tamara had asked me to share the experiences we've had in Texas addressing some issues that have popped up in DNA mixture interpretation. My talk will be not as long as the others. I want to make sure that there's plenty of time at the end because we have a lot of people on the line for questions. And so what I'm going to try to focus on is really the interaction between the legal system and some of these technical issues that have come up in DNA interpretation. So first, I'm going to talk a little bit about why Texas is different in terms of the way we approach crime laboratory oversight. And then I'll talk about our DNA mixture review project and some lessons that we've learned from that process. We've reviewed a lot of CPI cases, and I will talk about how that came about and why we decided that that was the right course for our state to take. We'll talk about the transition to probabilistic genotyping and some of the common courtroom challenges we've seen, understanding the tool, the probabilistic genotyping software as a tool, and speaking a little bit about its limitations, some things we've seen so far in practice in our laboratories. And then we'll talk about a little bit of a tension between Texas and SWGDAM guidelines. And specifically, we're going to focus on some subjects that our labs have brought to our attention at the commission, specifically the guidance around inconclusive ranges, the verbal scales or the verbal equivalencies, and weighted averages. So we're going to talk about how those have played out in Texas so far. So just in terms of framework, what do we rely on in the United States to ensure, to give confidence about the quality of DNA analysis that's happening in our crime labs? A big one is the accrediting bodies. The vast majority of laboratories, and certainly laboratories that are doing DNA work, are accredited by ANAB or A2LA or some other recognized accrediting body. Right now, most of our labs in Texas are ANAB labs. And I cannot tell you the number of times that you'll read a transcript in Texas, and I'm sure elsewhere, and the very first set of questions from the prosecutor typically is around accreditation and what it means. And typically, analysts, in order to simplify things, will talk about the fact that accreditation and quality go hand in hand. Sometimes we've even seen transcripts where analysts will talk about the fact that the FBI has approved all of our SOPs and everything we do because of the QAS. Now, we all know that that's not actually true. That's not the way it works. But what we've seen is sort of a historical reliance on those systems as a way to ensure quality. SWGDAM guidelines have been a huge place for analysts to go and for management to go to figure out what they need to be doing and what the latest consensus is in the community. And so SWGDAM guidelines are often pointed to as a place that help us understand where the community should be. And now we have the OSAC process. And that is promising to provide standards in place of what SWGDAM would release as guidelines to give even more teeth and meat to the process. Then we have the adversarial system, which is supposed to, in the U.S., the tug and pull and back and forth between the defense and prosecution is supposed to help ferret out a lot of these issues. and ultimately the judges act as the final gatekeepers of science. So here's the issue that we found with our commission is that oftentimes, if you ask the accrediting bodies, well, why didn't you identify some of these issues in mixture interpretation that were highlighted by the mixed studies? How come nobody ever flagged those things during an assessment? Well, they will tell you that they are not the scientific specifiers. They're not the ones who decide what is good or bad science in DNA. That's left to the community in the form of the SWIG-DAM guidelines. And if you talk to folks about why they didn't follow the SWIG-DAM guidelines at certain points in their history, they'll say, well, those weren't standards, they're guidelines, they're not rules. We did the best we could to get in compliance as fast as we could, but they are not rules, and we're not bound by them. So in the United States, what we've seen in some experiences at the Commission is that there are some gaps because a lot of the bodies that are in charge of various aspects of quality, when things don't go so well, sometimes we'll point at the other bodies as having dropped the ball. And so in Texas, what we have is a little bit of an extra layer of oversight in the form of our commission. It's not a perfect process. It's not a perfect body. No government body is. But what we do is sort of try to buttress the work of these other really important bodies like SWIG-DAM and like the accrediting bodies. We sort of do accreditation plus, and for those folks on the line who are familiar with our commission, who are analysts in Texas or lawyers in Texas, you know how it works. We see things because we receive complaints and self-disclosures from our laboratories. We see things that the accreditation assessment process may have missed. Now, if you talk to an assessor who's done this for a while, they'll tell you that they come on site for assessments for a set period of time, and they're supposed to accomplish a lot of stuff in that amount of time. And sometimes a big issue will pop up, and they'll have to spend a significant amount of their time dedicated to that issue. And it is possible that things will get overlooked in that process. And so we have the authority from the state of Texas, from our legislature, to go in and do audits if we so choose. If there's a red flag that's been brought up or an issue that we believe needs follow-up, we can go in and do that. So it's helpful to buttress the regular work that's done in accreditation assessments. The analysts in Texas are now licensed. They have licenses just like lawyers. And what that means is they go through a series of requirements that our commission has imposed, including they have to study for and take an exam that covers areas like confirmation bias or cognitive bias. They read about these issues. They read about their professional obligations under our Code of Professional Responsibility. And they're tested on it. And if an analyst were to commit misconduct, they can have their license taken away or suspended or probated, and this information is made public. Now, I can tell you that in the history of my time with the commission, I can count on one hand the number of instances where we've seen that type of misconduct that would merit a disciplinary action. But we're in a human endeavor. There are human beings involved in all of the work that's done. And it's very high stakes work. And so we do occasionally see misconduct. And what's nice about our process is we ensure that that analyst who may have dry labbed or done something else outrageous is not able to go to the county across the state and work for the laboratory there. In our state, laboratories self-report nonconformities to us. And that is a huge, huge advantage in terms of moving the laboratories toward transparency. We vet complaints in public meetings, we talk about the issues, and we make sure that the stakeholders who are interested or may have an interest in an issue receive information about what's going on. And the benefit of doing all of that, it may sound terrifying to have all of that put out there in the public, but what we're trying to do is foster a culture and a community that embraces mistakes when they get made as a point a jumping off point for learning and for progress. And striking the balance between accountability and sort of learning and embracing mistakes can be challenging, but we really try to do it. And the way we do that is through embracing certain values as an organization, as an oversight body. One is collaboration. Now, I wish I could have a system like what Dr. Gill described, where the scientists are forced to get in a room and figure things out and report those conclusions or recommendations about how things should proceed to the court. We don't have that system here, by and large. But because our commission is not in a court setting, we operate outside the adversarial system, we're able to be more collaborative than the court system in the United States allows in the context of any given criminal case. So that's a benefit, and we try to make sure that we take advantage of that in bringing all parties to the table to discuss issues. We try to be proactive, not waiting for something to kick us in the behind. When we learn about an issue, we try to tackle it and talk to the labs about the best and most efficient way to do that so that we don't paralyze them in the process. Transparency is a huge value, and it's because we've had a lot of exonerations in Texas. It's a painful process when you have to face that reality. And so our legislature has moved our state as far into transparency land as you can possibly be. Is it perfect? Do we get it right all the time? No. But I'm going to talk to you a little bit about the history of why we've ended up there. Blame-free is a huge piece of what we try to focus on. Now, there are circumstances when accountability is appropriate. When someone does something that violates the code of professional responsibility, when they take evidence from one case to report the results in another case because they're struggling with the data, that is not a blame-free scenario. That is a you need to not be doing this job anymore scenario, and we need to take your license away. But like I said before, that happens so infrequently. And when we see things, when we see issues in interpretation or problems in casework, it is very rarely and something that can be put squarely at the foot of any one examiner. Usually there are multiple factors going on as to why something did not go the way we had hoped. And so we try to focus on that as opposed to just nailing the analyst against the wall, which doesn't help anyone and doesn't get to the core issue that may be going on in the laboratory. We try to be solution-oriented. How can we fix things? And mindful of the mission. And by that I mean, this is serious business. Criminal cases and the results impact life and liberty. Every one of them. We are not making widgets. What we do is super important. And in Texas, we execute people. And so literally it's not just about liberty, which is extreme enough, but sometimes whether someone will live or die is at stake. And so we try to never lose sight of the seriousness of the work that forensic analysts do. So I just want to talk a little bit about our DNA mixture review project. I've talked about this at various conferences. If you've heard me speak at ISHI or the Bodhi conferences, I've talked about this over the last 4 years now. And so just as an introduction, back in 2015, a lot of you may remember this was all over the news at the time, that the United States Attorney's Office in D.C. asked Dr. Bedoli to consult on a homicide case that had been worked by the D.C. laboratory. And Bruce looked at that case and identified some concerns about the way the laboratory was interpreting mixtures using the CPI primarily. And at the time, Dr. Bedoli, as most of you know, is here in Texas with us. He is now a member of our commission. And so I called him and I said, I know the people, some of the people at that laboratory, and I'm just having a hard time wrapping my head around what's going on here with mixture interpretation. Do you think we could have similar issues in Texas? And he said, well, it's entirely possible because there's a lot of misunderstanding and confusion nationally about how to properly apply this particular statistical method. The CPI is what we were talking about. And so I said, well, let's get ahead of this. And so we brought all of our laboratories together to start talking about where everyone stands with mixture interpretation. And right around the same time, about a month later, the FBI released their amended STR population data set announcement. And in that process, the laboratories offered to perform recalculations for clients or customers, DA's offices, submitting agencies that had perhaps a pending case and they wanted to have a new result using the amended data sets. And everyone at the time expected that the impact of that would be insignificant. And so we all assumed that that's the way it would go. Except that if the laboratory had, say, originally issued a report in 2004 timeframe perhaps, and that report had a CPI stat, and the laboratory at that time had not yet incorporated a stochastic threshold, or they had incorporated one, but it was inappropriate. In other words, it perhaps was a quant-based threshold instead of an RFU-based threshold, so it didn't do much for the evaluation of stochastic effects in a mixture. And then the laboratory is asked to recalculate that statistic in 2015 after they had done a bunch of work on their mixture protocol to bring it in line with the Swigdam recommendations of 2010. If all that happened, and so then the laboratory accepts the request from the prosecutor to reinterpret the case, and they use their updated protocol to do that, you could see a significant change in your CPI statistic, or you could see a significant change even if that was run through probabilistic genotyping software for the reinterpretation. And that's a hard thing to talk to U.S. lawyers about, because typically the way lawyers react to changes in science or issues that pop up in science is one of two ways. It's either absolute panic, crisis mode, or they just say, I don't know what that means, just forget about it, I don't care. Just don't tell me about that, it's over my head. And so, as Dr. Gill said during his talk, we have got to move past that. The lawyers and judges everywhere I go, I talk to them about that they have to be able to handle these issues better. They need to become more educated if they ever hope to effectively try cases and to understand what is going on as technology advances. So we had a choice to make in Texas once we realized that some of these statistics were changing for those reinterpretations, changing beyond what had been expected just from the FBI amended data set announcement. What were we going to do? And so it was important for us, I think, as a state, to confront tough issues together. And I put this publication up here because anybody who hasn't read it, you really should read it. It just came out in fall of 2018, and it talks about the variation in mixture interpretation that was observed in the interlaboratory studies that NIST did, Mix 05 being one, and then Mix 13. What strikes me about this is that there is no place in the national rubric of oversight to deal with what is observed in those studies. In our state, we have a commission that said we're going to do something, but in most states, there's no place to go. And so what we choose to do here is to confront these issues together. And from our perspective, When you know that there's variation like that, or misunderstandings, historical ones that are not the fault of anyone, but that do exist, what should be done, right? We believe there's a duty to evaluate what laboratories were doing, to really ask, was it scientifically supportable? Were you using any of the urban legends that were described by John Butler in his book? Now, I want to pause for a second to talk about the Butler book, right? When we first started talking about these issues in Texas and the fact that the labs, some of them didn't have stochastic thresholds, or the stochastic thresholds that were set were not set appropriately, we got the reaction that, well, the first time anybody has said anything about this is John Butler in his book. Now, that's not true. Not true. Tamara put up examples of articles going back a long way, I think to 2000. I think Dr. Gill did the same, that the literature has been out there, but what we realized in this process, and I have a lot of empathy for the situation that the laboratories find themselves in. Dr. Butler summarized these issues in a nice, convenient little box, some of the challenges that and the myths that people were operating under and using that CPI stat. The literature was out there. He just summarized it in the box, which is great for everybody. But what dawned on me is, in this process, is there's a fairly significant gap between the amount of time and energy that folks who write the articles are able to spend on these complex concepts and what the analysts in the laboratory who are doing the work day in and day out have to spend. And right now we're in the middle of a legislative session in Texas, and I've been in hearing after hearing where the heads of our laboratories get reamed about the backlogs and the fact that there are so many sexual assault kits unworked and what a tragedy it is and what a crisis we're in. And those are not untrue statements. But when I go home, I wonder to myself, how much time do our analysts really have to read the literature? How much time do they have to go to conferences? And more importantly, not just go to the conferences, but when they get back, how does the learning that they are given access to, how does it translate into the laboratory environment? Are they given the time to do that? And I have to say that the gap that's there needs to be closed. It needs to be shrunk. And so we at our commission are really going to try to help with that process. Because it's critical. It's critical to make sure that we don't have to keep reinterpreting mixtures every 5 years because we realized that something wasn't quite right because people weren't given enough time to think the issues through. So when it comes to what needs to be done retroactively, if you were to reevaluate old cases now using proper methodology, would the stats change in a way that could be material to a defendant? And how do you know whether your answer to that question is correct or not? I believe, and our commission believes, and I believe that the laboratory directors from Texas have come to believe, as painful as it is, that this is necessary to preserve confidence in DNA analysis nationally. So here are some things I've heard in response to these ideas. The first argument being, well, Swigdam says the guidelines weren't meant to be retroactive, right? And I'm just taking this language as in every Swigdam document. It's not intended to be applied retroactively. Well, here's the thing. That depends. There's an element of a legal assessment that needs to be made in looking at that statement. We have something in Texas called Article 39.14 of the Code of Criminal Procedure. And what it says is that the state must disclose to the defendant any exculpatory, impeachment, or mitigating document, item, or information in the possession, custody, or control of the state that tends to negate guilt or would tend to reduce the punishment for the offense charged. Now, the information that a publicly funded laboratory has is considered part of the state's information in Texas and in most places. So for us, The answer to that swig-dam retroactivity statement is partially a legal analysis, and we try to keep that in mind. The language I just read to you is from a bill that was called the Michael Morton Act for the man you see here, because he spent 28 years in prison for having murdered his wife, and ultimately through DNA analysis, the Texas DPS lab discovered evidence that helped show that someone else killed Mr. Morton's wife and then went on, while Mr. Morton was about 19 months in prison, to kill another woman nearby in the same way that his wife had been killed. Mr. Morton sat in jail for 28 years. And when he was finally freed from prison, Texas took some aggressive action to expand our discovery rules and our notification obligations, because in Mr. Morton's case, some very critical evidence had been withheld that could have prevented his wrongful conviction in the first place. And I will tell you that what this quote from Governor Perry is absolutely on point when it comes to Texas. A lot of people are surprised by how, quote unquote, progressive our state seems to be on criminal justice issues. And it's just because of this. I mean, we are a law and order state, like I said before, and you cannot hold on to that reputation without considering the consequences and the responsibilities that go along with that. And so that's why in our state, we really think about how to make sure that things are fair and open. Other arguments I've heard, well, technology changes and we can't reanalyze cases every time. And what I would say to that is, look, it's absolutely true that if this was just a matter of going from one statistical method, the CPI, to probabilistic genotyping, we wouldn't be talking about reanalyzing cases. But if you know that you weren't applying the CPI statistic properly, that you were doing numbers matching or suspect-driven CPI or other unsupportable approaches to that stat, the answer to that argument is different. You have an obligation to fix those cases if there's any way that the statistical weight that was conveyed to the jury and the trier of fact is no longer accurate in a way that could be material to the case outcome. Those are the real key questions. And the other argument that we heard when we started embarking upon this reinterpretation process was, oh, don't worry, the numbers will just go up with probabilistic genotyping. Well, yeah, in many cases that's true, but not in all the cases, which we found because we just went through this process. So how do you even go about reanalyzing cases? It just sounds crazy. Why would anyone do that? Well, we have been able, through a collaborative process and a series of grants, we've been able to triage our caseload, the lawyers in the laboratories working together to identify, to narrow the number of cases that require reanalysis to make that process manageable. And so is it a perfect process? No. Do we know that we got everybody who needs attention? No, we don't. But we set up a process to go down that path, and then anyone else who raises cases to our attention, we will look at them. And Texas is happy to help any other state that considers, or any other jurisdiction, a smaller area, county, that wants to tackle a similar project. So here are our numbers, just to let you know. Since we started this in 2015, the laboratories have identified approximately 40,000 DNA mixture cases. Now, obviously, not all those went up into CODIS, but these are cases that they know could have had mixture evidence in them. And what they did was they gave those case lists to the prosecutors. Now, we have 254 counties in the state of Texas, and we don't know for sure that all of them sent notice out. But we do know, based on what we've seen in terms of responses, that at least 160 of them have. And we also posted notifications in the prison libraries all across the state, which Judge Hervey from our Court of Criminal Appeals, who some of you may know, she insisted on that. And so as a result, in 3,000, I think this number might be up a little bit closer to 3,500 at this point, but that's the number of people who asked that their case be looked at again. We have 351 cases still awaiting review by the lawyers, and the way the process works is the request for reanalysis comes into a team of attorneys. They evaluate the overall case, and they ask a fundamental question. If the DNA were taken out of the equation, if the DNA were removed from the case, or if this person had been excluded from as a contributor, would the case outcome have changed? If the answer is no, because the other overwhelming inculpatory evidence would just still be there and therefore the person would have been convicted, the case does not get reinterpreted by the lab. We're not trying to go down a path of reinterpreting old cases for the sake of it. We're trying to get to the ones where there may have been a miscarriage of justice, potentially, because the other evidence was not very strong and the DNA could have mattered. And so of all those cases that were looked at, 85% of them got closed by the lawyers because they said, we're not going to ask the lab to reinterpret this one. It doesn't merit reinterpretation. So the labs have been asked to calculate about 150. 30 of them have not yet been recalculated. Those requests are pending. And we've seen in about 6 cases that were put through StarMix after the original analysis, differences in outcomes that change to the defendant's benefit in a significant way, where those cases will need to be potentially relitigated, or at least an attorney will have to be appointed to represent those defendants. So here's one example of the way that those urban legends that are featured in that CPI box can play out in real casework. Now, I want to absolutely qualify this by saying these are not your typical cases. This is very unusual to see big changes like this. So that's the good news. But this particular case involved an aggravated robbery in 2010, and it was committed by 2 assailants who concealed their identity. It was the robbery of a home. And a white T-shirt was used to tie up the complainant. And that white T-shirt was tested to see what DNA results would come. And the report was initially released in 2011, and the result was that the defendant could not be excluded from a mixture found on the T-shirt, and the probability of selecting a person at random who could be a contributor was 1 in 87 for Caucasians. Defendant was a Caucasian. So that is a weak statistic, but it's still an inclusion, right? So the results were and the prosecutor, just to be abundantly clear, introduced this evidence at trial, did not decide he wasn't going to use it because the stat was pretty weak. He went ahead and used it, which I think many, many prosecutors in this state would have done. The results were reevaluated using StarMix in 2017, and the defendant was excluded. And so you might ask, well, how does that happen? And the answer was, if you look back at some of the issues that were common misconceptions, so the laboratory did not at the outset determine the number of contributors by evaluating peak-height ratios. They didn't think that was necessary for a CPI statistic. The SOP at the time said that all data above the analytical threshold established by the laboratory's validation were permissible to use. There was no stochastic threshold in place at the laboratory at the time the case was analyzed, and it was 2011, shortly after the Swig-Dam guidelines that talk about stochastic threshold were released. But keep in mind, the literature was there before. It's just that this lab, like many others, had not yet validated a stochastic threshold. And no real consideration, when you look at the profile, we know that there wasn't consideration given for the potential of allele dropout or missing data that could render a CPI inappropriate for this case. The reference sample of the defendant was compared, and the loci where the suspect's alleles were observed were used in the CPI stat. And where the suspect's alleles were not there, and there was another allele there that couldn't be explained, the explanation given by the lab was there must have been a fourth unknown contributor. Except that if you looked at the peak height ratios and you looked at the data, 4 contributors was not a supportable explanation for what was going on with that data. And so this is sort of how those urban legends can play themselves out and get you from a place where a CPI statistic originally calculated gets sort of turned on its head to an exclusion once the analysis is run using probabilistic genotyping software. And again, these are not typical, but when they happen, they matter, right? That result, going from any type of inclusionary type number, as weak as many would consider that to be, to an exclusion, that is Brady material. That is Michael Martinak material, without a doubt, because it is potentially exculpatory. Now, in that particular case, there was other inculpatory information or evidence linking that suspect to the offense, but now the lawyers are going to have to find out whether that was enough to sustain the conviction or not. So in this process of evaluating mixtures using CPI, as with the rest of the country, we started learning about a better way, and that's probabilistic genotyping. And the scientists, Dr. Gill and Dr. Moretti, will do a way better job of explaining why that probabilistic genotyping is such a better approach. But for my sort of limited lawyer brain, we know it makes better use of the data because you don't have those sort of cutting off things to avoid missing data resulting in losing good data. It removes the need for a stochastic threshold, and so to a certain extent also reduces subjectivity. But it's not a replacement for analyst judgment. It just isn't. And we see that in real practice, looking at recent examples of how the analyst still must stay engaged, still must make important calls. And if that doesn't happen, we can end up repeating some of the same mistakes that have happened historically. So just to give a little bit of very basic legal background about how an introduction of software like StarMix is considered in our adversarial system. In Texas, admissibility is determined under a decision called Kelly versus State, which is very similar to the Daubert decision at the federal level. Texas Rules of Evidence 702 sort of articulate the standard, and to put it in the simplest terms, the science has to be good foundationally. The scientist has to be competent to do the analytical work that's being offered. And in that particular instance of the case before the court, the application must be appropriate. That's how admissibility is evaluated when you just distill it down to the bottom line. Now, in Texas, we've had a lot of turnover with our judiciary, a lot of diversity in terms of new, younger judges, fresh faces coming in. In Harris County, which is Houston, 13 out of 23 new criminal district judges, they're brand new. 13 out of 23. That's a lot. And so with this exciting change in the judiciary, because our judges are elected, that also requires a significant amount of education. Judges have to get caught up on a lot of these concepts that aren't the easiest to explain. So in Texas, when we talk about star mix in particular, at the trial court level, star mix has been admitted in dozens of cases. And interestingly, because Texas DPS, our state lab, was the first laboratory to roll out star mix, and because they service a lot of the rural, smaller county populations in this state, where we have 254 counties, a lot of the admission of star mix evidence has happened in smaller counties first, not the big urban areas. At the appellate court level so far, we haven't had any cases that rule on admissibility of star mix. It has been mentioned, probabilistic genotyping software generally has been mentioned in 3 cases. That's a picture of our Court of Criminal Appeals. They will, I'm sure, issue a decision on this soon, but it has not happened yet, so we're still waiting on that. Now, at trial, the types of issues that have been seen by litigators, things that come up in the back and forth during admissibility hearings are, for example, whether the analyst can explain deconvolution of mixtures, the likelihood ratio generally, we're going to get into that, and specific challenges to star mix. And I probably should put that same disclaimer. I mean, we don't endorse any particular software. We make no comment about software generally. I will say that it just so happens that in Texas, our laboratories are all moving towards star mix, so I talk about star mix a lot. But that is a choice that's made at a laboratory level based on the needs of the lab and their procurement rules and so forth. So what we've heard on the subject of star mix admissibility in particular is that the software is a black box. What about programming errors? How do we know there weren't any? How do you address those? And then again, on the subject of the likelihood ratio, we'll talk about that in a little bit. They like to talk about the variability between runs, the fact that there are competing software programs, and Dr. Gill talked about that case example where 2 different programs yielded different results. Unfortunately, here, there are no joint statements forced upon the experts. So it's basically fought out in an adversarial setting. Probably not the best place to decide scientific issues, but that's what we're left to contend with. And then a lot of questions about whether the validation studies that are done really reflect the low-level degraded samples that are often seen in casework. So the one area we focused on a lot is the concept of star mix as a black box, where the analyst just inputs the data and it spits out an answer and that's it. But I would ask, is that really true? Because from what I've seen, that's not the way it should work. And if it does work that way, there's a problem. The analyst should not just be pushing the button and taking the answer. The analyst should be able to explain, maybe not the source code because they're not programmers, but they should be able to explain and understand the fundamentals of what it is that the software does. SOP should allow for an intuitive assessment of the mixture to even override the star mix result where appropriate. And Tamara talked about those issues in her talk. And it's especially important when it comes to likelihood ratios down near 1, where a holistic review of what's going on in that mixture is really merited and warranted. We've seen some examples, or at least an example recently in Texas, where the assessment of the number of contributors can really matter, right? So if you overestimate the number of contributors, say you call a mixture 3 persons instead of 2, it can lead to a slightly inflated likelihood ratio. And these megaplex kits, from what I understand, and I'm saying this in the simplest possible way, being a lawyer, but there's greater peak height ratio variability than what used to be in the kits where you were evaluating 13 loci, and now that we're moving to these big kits, there's greater variability. And so how you do your validation study when it comes to those questions around peak height ratios for assessing the number of contributors becomes really important. Because in the example of one laboratory that we've seen recently, if you call something that should be a 2-person mixture a 3-person mixture, it's possible that your likelihood ratio is going to move from an exclusion over into a weak likelihood ratio, like 0.017, say, for example, or 0.015 or something. And for a laboratory that has established an inconclusive range that, say, is, anything beyond 0 into, say, 100 or 1,000 or whatever that inconclusive range is, you're going, for all intents and purposes, for what you're communicating to the trier of fact, you're going from an exclusion to inconclusive. And if you talk to any lawyer in this state, if you ask them, would it matter to a jury whether the answer is that the defendant is excluded as a potential contributor or that the assessment of the evidence is inconclusive, would that matter? And the answer, I will tell you, is a resounding yes from the lawyers that we talk to. And so these types of things play themselves out in actual casework in a pretty major way. So there is no way to move to a probabilistic genotyping software and leave your brain at the door and forget all of these things. These concepts, the analysts are responsible for being the expert. They are the expert, not the software. So moving along to this idea that I introduced a little bit at the beginning, Texas and the federal government, we have tension around various issues. That is probably not a surprise to anybody. And the SWGDAM guidelines are no exception. Now, generally, we're in support of the guidelines and all of the very, very critical, important work that the SWGs do, and including SWGDAM. And the OSAC, and these are all great initiatives, and Texans are involved in all of it. It's not to say that we're just sitting down here on an island. But there are a few areas of disagreement that the labs came to us about when the most recent guidelines were issued. One of them was on the question of weighted averages, and from what I understand, the recommendation essentially says that a likelihood ratio that combines information from different population groups may be used in lieu of the single lowest value. We don't agree with that approach because we don't know the population stratification of the area where the crime was committed. For example, there are some parts of Texas along the border in the valley that are 95% Hispanic or Latino, and we don't want to skew the results in any way that is problematic based on the reality of those population groups. What if the crime's committed in multiple locations? And so, in our view, it's better to use either the lowest value or to report all the populations that are routinely considered and let the trier of facts look at that and make a judgment about what is most appropriate. So that's one example of an area where we differ. The second one, which is probably the bigger one, is about verbal qualifiers or the verbal scale. And everybody knows what it is. It's sitting there to your right. Now, when this was introduced, a very brave soul from Texas DPS, an analyst, a DNA analyst who was involved in StarMix validation, got up in front of a room full of lawyers and said, we're thinking about adopting the verbal scale. And they pounced on him and basically said, what are you doing? Words are our province, our being the lawyer's province. Those words, you should not be assigning words to the numbers. That's for us to do. They also hated the concept of a prosecutor's hypothesis and a defense hypothesis. They wanted us to use hypothesis 1 and 2 or A and B or anything else but those words. Now, if you talk to Ted Hunt or Tamara or a whole bunch of people that were involved in the creation of these verbal qualifiers, one of the big points that they talk about in favor of adopting a verbal scale is consistency. We can give the same message and be consistent. We should do that, right? To eliminate or reduce the variability between the words that people are using when they get on the stand. And that is an absolutely valid concern. And it happens, right? If you read transcripts, you can see it. And then the question, though, in response to that is, do we really know what juries hear when they hear the words moderate or strong or very strong? What does that mean? Or should we let the lawyers assign those descriptors and find a different way to express the likelihood ratio and to try to give it some context? I'm not saying we have landed upon a better way, and I'm sure a lot of people will argue with something that we're floating, which I'm about to talk about, but we think we still need to work on this concept before going with the verbal qualifiers that are proposed. So one way to do it is to provide an appendix to the DNA reports that state, in as plain terms as we can, what a likelihood ratio is, and then describe what we talk about when we mean that we are essentially evaluating how well two different explanations account for the DNA profile observed on an item of evidence, and explaining that sometimes and often the state has one explanation and the defense has a different explanation. And in this idea draft that we're floating, we would refer to the first explanation as the inclusion explanation and the second alternative as an exclusion explanation. And then we try to explain to the jury that the size of the LR indicates the relative strength of support for each of these explanations of the evidence or the evidentiary data that the laboratory looked at. So here's an example. I'm just going to pause for a second, because when I was putting this together using guidance that I got from Dr. Tim Slider, who's at the Dallas County Lab, I kept trying to simplify the language that he had given. And what I realized in a panic was that in doing that, I defaulted to the prosecutor's fallacy. And then I had to redraft it and pray that I had gotten it right when I went back to try to fix it. It is so easy for lawyers in their attempt to try to write language that is simple for a jury to screw it up. Now, I hope I have not done that here, but here's an example of the type of chart that we're thinking about providing. So the first row describes the evidence. The evidence is a bloody knife recovered from a murder scene. The data from the bloodstain indicates a mixture of DNA from two people. So we're saying what it is that the laboratory is evaluating. It's a mixture of DNA and how many people are believed to be contributors. And then we say the exclusion explanation that we talked about in the appendix would say the following, that the evidentiary data observed is more likely explained, better explained as a mixture of DNA from the victim and the suspect, where the alternative explanation to that, that probably is being proffered by the defense, but not necessarily, the alternative would say that the evidentiary data observed is more likely explained as a mixture of DNA from the victim and another person who's not the suspect. And then if we have a likelihood ratio of 100, we put the number right there, or whatever that number is. We issue the conclusion. The DNA profile from the bloody knife is 100 times more likely to be observed if the victim and suspect contributed the DNA than if the victim and a random unrelated person contributed the DNA. And then we talk about meaning, context, and we would say that an LR of 100 means that in a population of people equal to the population of Texas, approximately 283,000 would be expected to give an LR greater than what was reported here. And then we have the data, the tables that show the population of Texas, the United States, and the Earth to give context. And we're even thinking about introducing another column that would be the population of whatever county or region of Texas the crime occurred in. So that's one way that we're thinking about presenting the data that doesn't rely on these words here, uninformative, limited, moderate, strong, and very strong, but rather gives some context that we think most juries will be able to appreciate. So we're still talking about that, and but the labs really are not quite comfortable with using the verbal equivalency just based on feedback they've gotten from the legal community. And then finally, one issue that we have talked about is the language in the SWGDAM guidelines that talks about inconclusive ranges. And what it says is that a likelihood ratio appropriately conveys the weight of the evidence and shouldn't be reported as inconclusive based on its magnitude, right? So we agree with that in principle. An analyst should be able to explain what a low likelihood, say, of 100 means. But our thoughts, and this is largely something that Dr. Badoli has talked about at UNT, is that there could be a good use for an inconclusive range in a different way, where if you have a single DNA extract and it's low level, and you aliquot it again and again and again and again, and that LR fluctuates in some way every time you run it, through validation, you might, as a laboratory, want to develop an inconclusive range for that type of scenario. And so I think UNT is working, Dr. Coble and Dr. Badoli, now on running those samples to see what happens, and then to make a decision about whether an inconclusive range might be appropriate for those types of samples and scenarios. Now, I just wanted to let people know that if you really want to hear more, we did a 2-day symposium at the University of North Texas where Dr. Buckleton was there, Alan Jameson, David Kay, Tamara, Joe Bright, Bruce, Mike Coble, lots of people who know lots more than I do about all of these issues. And there was great debate about various developmental validation issues, laboratory validation, all kinds of questions came up about the SWGDAM guidelines, the verbal scale, etc. You can watch those that symposium, and I've put the links in here. A lot of people are using it as a training tool, and I think it's a really great one. And we would encourage you to take advantage and watch it whenever you have some time. And that's all I have. Thank you to all these folks that helped with the presentation. And I don't know if we still have time for questions, but I'll stop sharing my screen and turn it back over to Tamara.
Okay, thank you so much, Lynn. We just have a few minutes remaining, but I think perhaps a couple of them you could answer really easily or quickly for us. Who is in charge of licensing forensic analysts in Texas?
Our commission licenses the analysts. We do it.
Okay. Okay. And what's involved in that licensure?
So there's an educational requirement, minimum coursework, there's proficiency testing, there's an exam that they have to take, continuing education. There are quite a few items. A statistics course now, everyone has to take stats, which is easier for the DNA folks, but is causing a little bit of consternation among the other disciplines. All of those things are involved.
Very good. Are the defense experts also required to be licensed?
So the way the law works right now is that people who work in an accredited laboratory must be licensed. So, for example, Bodie, Sorensen, NMS, all those private labs, if they do any Texas casework, whether it's for the defense or for the state, they are all licensed. Even though they're not physically located here in Texas, they're licensed.
Very good. The first question up there, I think we're going to we'll have a whole webinar directed on the SWGDAM scale and other ways of communicating the results. So we can come back to the topic of inconclusiveness at that time. Let's see if we can talk about the next question. Is the commission concerned that an LR at activity as was quoted by Dr. Gill at 12 to 40, in a situation when the subsource was on the order of 1 billion?
So I'm going to be totally honest on this question that on the issues that Dr. Gill presented about subsource versus activity level that I know of, and Tamara, correct me if I'm wrong, but I don't think our labs do that type of analysis on the stand. They report their results, the likelihood ratio at the subsource level, and I'm not sure that they go on to talk about the impact of other facts in the case on the likelihood ratio. Does that make sense?
Yes, it does. You're right. Do you think that laboratories, especially those laboratories that have backlogs, will actually have the resources to carry out experiments along this level? Or are we looking to some other body perhaps to address these issues from a research perspective?
There is no way right now that our laboratories could do that work. No way. Except for probably the University of North Texas. Our laboratories have all they can do to catch up with the demands that are being placed on them by their submitting agencies. I would love for our labs to be able to get involved in that type of research, and I hope that we're going to be able to find a way to do that, and I think Bruce is really going to try to work toward that goal. But right now, the reality, at least in Texas, is that no, they've got all they can do to keep their heads above water. That's just the reality.
Okay. Well, Lynn, thank you so much for your talk. In fact, your issues at trial that you described, from the black box to numbers of contributors, when to override the results, being the expert, trying to simplify the expression, and so doing, committing the prosecution fallacy. These are all topics that we are on schedule to cover in the next couple of months. So I would encourage everybody here today. I think we had 499 logs in on last count, to spread the word, and let's try to get some more people and welcome you all back next week at the same time, when we're going to be talking about statistical aspects of probabilistic genotyping. We have David Balding, Michael Coble, Stephen Myers, and John Buckleton at that order of appearance. So hopefully we will meet with you all again at the same time next week. Thank you so much for coming, and I want to pass the mic over to Josh to see if he has any concluding comments for us. Thank you.
All right, thank you so much to Dr. Moretti, Dr. Gill, and Lynn Garcia for their presentations today. As Dr. Moretti said, we will be having another workshop in this series next Wednesday, so be sure to register for that. That way you can continue along with the workshop series. We also would like to hear your feedback about what you thought, not only about today's event, but also the new platform. If you scroll down below the captions, there is a button there that once we are finished, you can click on for the survey. So simply click on that. It's a simple 2 to 3 minute survey. We would greatly like to hear your feedback about our delivery today and the new platform, what issues you did run into, and how we can better your experience for our next webinars and next week. We are also going to be putting some information there in the chat pod about our social media outlets, such as Facebook, LinkedIn, and Twitter. If you are not following us on any of those, please feel free to look us up on our social media and follow us from there, as well as our podcast and our website will be put up there in the chat pod as well. We did record today's event, so we will be converting that into our archival version over the next week, and then we'll get that back out to you, our audience, so that you can reference that. Again, thank you so much for everyone attending. This was a massive event and a massive success. I know that some experienced problems, but as Dr. Moretti said, we did have almost 500 that attended today, so that is fantastic for for the Forensic Technology Center of Excellence and the forensic community as a whole. Again, thank you so much for attending today, and we look forward to seeing you in the next workshop in the series. We hope everyone has a great afternoon.